Hello and welcome back to Secrets After Midnight. Tonight we are going to spend some time with two of the most despised creatures on Earth, and by the end, you may never see the natural world the same way again. The cockroach and the beetle. Between them they have outlasted ice ages, continental collisions and 5 mass extinctions. They were here when the first forests rose from bare rock. They were here when the dinosaurs fell. And right now, at this very moment, they are doing quiet, essential work inside every functioning ecosystem on the planet, recycling the dead, aerating the soil, pollinating ancient plants, and breaking down waste that nothing else will touch. We call them pests. Science calls them irreplaceable. This is not a story about disgust. It is a story about misunderstanding, about what happens when we judge a creature by how it makes us feel rather than what it actually does. Over the next 100 facts, we are going to move through the physical architecture of these animals, the surprising depth of their minds, their rich social lives, their place inside human history, and finally, the quiet stillness of how they rest when the world goes dark. Some of what follows will be hard to believe. All of it is real. 300 million years ago, before the first dinosaur drew breath, before a single flower had ever opened on Earth, a creature moved through the undergrowth of a vast carboniferous forest. Its body was flat and wide. Its antennae swept the air in slow arcs. Its legs were built for speed across uneven ground. That creature was in all meaningful ways a cockroach, and it looked almost exactly like the one that might be living behind your refrigerator right now. No other animal on land has held the same fundamental body plan for that length of time without significant revision. Not sharks, not crocodiles, not horseshoe crabs. The cockroach represents what biologists call a persistent morphology, a design so precisely fitted to survival that natural selection has found no reason to improve it across 300 million years of relentless environmental change. That is not stagnation, that is perfection. The flat, low profile allows it to compress into cracks. The wide-set legs provide stability at speed. The long, flexible antennae sample air chemistry with extraordinary sensitivity. Every feature has a function. Every function has lasted longer than the mountains of Europe, which did not exist when this body plan first appeared. A cockroach can survive without its head for several weeks, and this is not folklore. It has been documented, studied, and replicated in laboratory settings with careful controls. The reason is architectural. A cockroach does not rely on its brain to regulate blood pressure, breathing, or basic movement. Its nervous system is distributed clusters of nerve tissue, called ganglia, run the length of its body, each one capable of coordinating the limbs and organs of its local region without instruction from above. Breathing in a cockroach happens through spiracles, small openings along the sides of the body that open and close independently. No brain required. The headless body simply continues, responding to touch, moving its legs in coordinated patterns, maintaining respiratory function. What eventually ends its life is not the loss of the brain, but thirst without a mouth it cannot drink. The head, meanwhile, fares slightly worse. Severed, it will continue waving its antennae for hours, responding to chemical stimuli, twitching in patterns that suggest the nervous tissue remains active. Scientists still debate the precise duration and mechanism of this post-decapitation activity, but the fundamental reality is not in question. The cockroach body is less a single organism than a confederation of systems, each one resilient enough to operate alone. The cockroach exoskeleton is made of overlapping plates, connected by flexible membranes, a design that allows the body to compress vertically to nearly half its resting height without any damage to the internal organs. Researchers at the University of California, Berkeley tested this using controlled compression rigs and high-speed cameras, and what they found challenged the assumption that rigidity equals strength. A cockroach walking through a gap so narrow it seems impossible does so by splaying its legs outward and allowing the thorax plates to slide over one another like roof tiles in a wind. The internal organs tolerate this compression because they are suspended in hemolymph, the insect equivalent of blood, rather than fixed in position. The result is something like a biological accordion, soft enough to reshape under pressure, structured enough to spring back completely. Think of squeezing a full deck of playing cards flat, then watching it restore its own shape the moment pressure releases. Engineers at Harvard and Berkeley have begun modeling cockroach-inspired robots using this principle, building machines that can slip under doors, navigate rubble, and compress into spaces far smaller than their resting profile. The cockroach did not invent flexible armor, it perfected it. 250 million years before we thought to ask, the American cockroach times, Periplaneta Americana times, can reach a speed of roughly 50 body lengths per second at full sprint. To put that in terms, a human body can feel if a 6-foot person moved at the same body length ratio, they would be covering ground at close to 200 miles per hour. For a brief moment during peak acceleration, the cockroach actually rises onto its 2 rear legs and runs bipedally, leaning forward like a sprinter at the starting block. This was captured on high-speed film and initially met with skepticism. Bipedal locomotion in an insect seemed unlikely, but the footage was unambiguous. At top speed, the front 4 legs lift entirely off the ground. The transition happens in milliseconds and is triggered not by a conscious decision, but by the physics of acceleration itself. As speed increases, the rear legs generate enough forward thrust that the front of the body rises to maintain balance. The cockroach does not choose to run on 2 legs. Physics chooses for it. This biomechanical quirk has been studied extensively by robotics researchers because the stability the cockroach maintains during this transition, without falling, without losing direction, represents a control problem that no wheeled or legged robot has fully solved. The cockroach solves it every time it flees a light. Along the lower legs and abdomen of a cockroach there are clusters of tiny sensory hairs, each one connected directly to a nerve fiber, each one tuned to respond to specific frequencies of air displacement. These hairs can detect the movement of air caused by an approaching predator before that predator makes audible contact with the ground. The system responds in under 10 milliseconds faster than the cockroach's own brain can fully process the signal. What happens is essentially a shortcut. The sensory hair triggers a motor response in the local ganglion, which initiates the escape run before the brain has received and interpreted the signal. The cockroach, in other words, begins running before it consciously knows why. This is not instinct in the vague colloquial sense. It is a precisely engineered reflex arc, a piece of biological hardware built for one purpose. Researchers studying these hairs found they are directionally specific hairs. On the left side of the body are more sensitive to air movement from the left, which allows the cockroach to automatically orient its escape away from the source of the disturbance. You may have noticed, if you have ever tried to catch one, that a cockroach seems to dodge in exactly the wrong direction for you to grab it. That is not luck. It is geometry. At the rear of the cockroach's abdomen, 2 short appendages called cerci extend like narrow antennae pointing backward. Each circus is densely covered in filiform hairs, and together they function as a seismic detection system of extraordinary precision. Laboratory measurements have shown that cerci can detect substrate vibrations, tremors traveling through the ground or floor at amplitudes too small for most mechanical instruments to register without amplification. Each hair on the circus is mechanically tuned to a specific frequency range, so the system does not merely detect vibration but characterizes it, distinguishing, for instance, between the footfall pattern of a spider and the vibration of a passing truck. Scientists still debate whether cockroaches use this information to construct a spatial model of their environment, or whether the response is more reflexive, a detection to escape pipeline without conscious analysis. What is not in question is the sensitivity itself. The cockroach, resting still in darkness, knows through its hind end sensors that something is moving nearby before any other signal reaches it. If you have ever entered a room at night, turned on a light and watched a cockroach vanish before you thought you had made any noise, the cerci are the reason. It felt the pressure wave of the door. A cockroach breathes through a series of small openings along its sides called spiracles, and unlike the continuous breathing rhythm of a mammal, it opens those spiracles only in controlled, intermittent bursts. For stretches of up to 40 minutes, a cockroach can keep all its spiracles sealed, halting gas exchange entirely to prevent water from escaping through the respiratory system. This strategy is called discontinuous gas exchange, and in arid environments, it represents a survival adaptation of remarkable sophistication. Water loss through breathing is in dry conditions a genuine threat to insect survival, and the cockroach manages it the way a careful traveller manages fuel. Open the valve only when necessary. Close it as soon as possible and measure the cost. Every time. During the closed phase, carbon dioxide builds up in the body tissues, and oxygen depletes slowly. When the threshold is reached, the spiracles open, briefly flush the system, then seal again. The cycle repeats, according to the body's metabolic demands, not a fixed clock. Researchers have noted that this is one of the reasons cockroaches can survive for long periods submerged, not because they breathe water, but because they were barely breathing to begin with. The cockroach, it turns out, holds its breath far better than most creatures that evolved specifically to swim. A cockroach has no veins, no arteries, no capillary network threading through its tissues the way blood vessels thread through a human hand. Instead, its body cavity is filled with hemolymph, a pale fluid that functions as blood, lymph, and interstitial fluid simultaneously. And that fluid is moved not through sealed tubes, but pumped by a long dorsal vessel running along the top of the body, then released directly into the open cavity where it washes over every organ at once. This is called an open circulatory system, and it appears primitive until you consider what it actually accomplishes, because every organ is bathed directly in hemolymph. Rather than relying on capillary delivery, there is no single bottleneck, no main artery, whose blockage causes catastrophic failure. The system has no equivalent of a heart attack. Pressure drops fluid, redistributes organs, adjust. The hemolymph carries nutrients, hormones, immune cells, and crucially, antimicrobial peptides that have attracted serious attention from pharmaceutical researchers. Several compounds isolated from cockroach hemolymph have shown activity against bacteria resistant to conventional antibiotics, including some strains of MRSA. The creature we spend billions of dollars trying to kill may in its blood carry chemistry capable of saving human lives. That particular irony is not lost on the scientists doing the work. The hemolymph of the American cockroach contains at least 9 distinct antimicrobial peptides, small protein molecules that punch holes in bacterial cell membranes, destroying pathogens from the inside out. Researchers at the University of Nottingham isolated several of these peptides and tested them against some of the most dangerous drug-resistant bacteria known to clinical medicine. The results were striking enough to prompt continued research. Certain cockroach-derived compounds eliminated more than 90% of MRSA bacteria in laboratory conditions and showed similar activity against Escherichia coli, strains that no longer respond to standard antibiotics. The cockroach lives in environments saturated with bacteria, fungi, and decomposing organic matter. Its immune system did not evolve for a clean laboratory, it evolved for a sewer, a compost heap, a crack behind a hospital wall. That relentless microbial pressure, sustained across hundreds of millions of years, produced a biochemical toolkit of extraordinary sophistication. Scientists still aren't sure exactly how the cockroach deploys these peptides internally, whether they circulate constantly or are produced in response to specific threats, but the compounds themselves are real, measurable, and irreproducible. The cockroach we spray with poison may be carrying in its pale blood the next generation of antibiotics. Several cockroach species are capable of parthenogenesis, reproduction without fertilization, in which a female produces offspring from her own unfertilized eggs. In the American cockroach, this occurs occasionally and produces viable nymphs. In the Asian cockroach, it has been documented as a regular reproductive strategy under certain conditions. What makes cockroach parthenogenesis particularly remarkable is not simply that it happens, but that the resulting offspring are not clones in the strict sense. Genetic recombination still occurs during egg formation, introducing some variation into the population. This means a single female, isolated from all males, can establish a genetically diverse colony over multiple generations. In practical terms, this is part of why cockroach infestations are so difficult to eliminate. A single pregnant female or a single female capable of parthenogenesis is sufficient to repopulate an entire building. Entomologists studying pest control have noted that eradication programs which eliminate 99% of a population may still fail, because the surviving 1% contains individuals whose reproductive strategy does not require a partner. The math is unforgiving. It connects quietly to something most people have experienced, the unsettling return of a problem you were certain you had solved. The German cockroach Blatella germanica is the most common indoor species worldwide, and its reproductive capacity borders on the arithmetically surreal. A single female produces an egg case called an ootheca approximately every 6 weeks. Each ootheca contains between 30 and 40 eggs. The nymphs that hatch reach reproductive maturity in roughly 60 days under warm conditions. If a single female's offspring survive and reproduce without interruption, which does not happen in nature but illustrates the biological ceiling, the theoretical population after 1 year exceeds 10,000 individuals. Under real-world conditions, accounting for mortality, competition, and resource limits, documented infestations have grown from a single breeding pair to several thousand individuals within 4 to 5 months. This is not a design flaw in the cockroach. It is a design feature, an evolved response to the enormous predation pressure these animals faced across hundreds of millions of years. Most of those offspring die. The strategy is not efficiency but volume. Produce so many that survival even at low rates guarantees continuation. It is the same logic that plants use when they release 10,000 seeds to grow one tree. The cockroach simply does it faster and with more legs. The ootheca is one of the more elegant structures in the insect world, produced by the female cockroach from a protein secretion that hardens rapidly on contact with air. It is a small symmetrical capsule ribbed along one side like a tiny leather purse that houses between 16 and 50 eggs arranged in 2 neat rows. The outer wall of the ootheca is chemically treated as it forms, with a compound that repels water on the outside while maintaining humidity within. This creates a microenvironment inside the case that remains stable regardless of whether the surrounding air is dry or damp. The eggs inside do not breathe in the way adult insects do. They absorb oxygen through a controlled diffusion process across the case wall, which is porous at a microscopic level, but sealed against liquid water. Different cockroach species handle the ootheca differently. The German cockroach carries hers attached to her body until hours before hatching, protecting the eggs with her own body heat and immune chemistry. The American cockroach deposits hers in sheltered crevices, often gluing it to a surface with a secretion. Some species chew the ootheca into the substrate entirely. The variation in strategy across species reflects the different environments in which each evolved, but the engineering of the case itself is consistent, refined, and extraordinarily effective. Inside, the cockroach gut lives a community of microorganisms so metabolically diverse that entomologists sometimes describe it as a miniature ecosystem in its own right. Among its most ecologically significant functions is nitrogen recycling. Cockroaches feed on material that is often nitrogen-poor, dead wood, paper-decaying plant matter, and yet their bodies maintain adequate protein levels. The mechanism involves symbiotic bacteria in the gut that fix atmospheric nitrogen and convert waste nitrogen compounds back into usable amino acids. This means the cockroach partially feeds itself through its own microbiome, extracting nutritional value from its waste before discarding it. In forest environments, this makes the cockroach a genuine contributor to the nitrogen cycle, breaking down material that would otherwise lock nitrogen away in inert form and returning it to the soil in bioavailable compounds that plants and fungi can use. Remove cockroaches from a closed forest system, and measurable changes in soil nitrogen content follow within years. Researchers studying tropical deforestation have noted that cockroach population collapses in disturbed forest patches correlate with detectable reductions in soil fertility. The creature we associate with filth is in the ecosystem that built it. A fertility engine, small, tireless, and chemically ingenious. The cockroach digestive system produces a suite of enzymes that mammalian biology simply does not possess. Chief among them are cellulases, enzymes that break down cellulose, the structural material of plant cell walls, and keratinases, which dissolve keratin, the protein that forms hair, feathers, and fingernails. A cockroach can extract caloric value from a leather shoe, a cotton shirt, or the glue binding the spine of an old book. It can digest human hair clogs from a drain process, the dead skin cells that accumulate in a mattress, and extract nutrients from the paste used to hang wallpaper. None of this is opportunism in the negative sense. It is ecological function. In a forest, these enzymes are deployed on fallen wood, shed fur, Dead leaves the vast reservoir of complex organic material that would otherwise accumulate faster than it could be broken down. Every cockroach feeding on a dead log is performing the same biochemical work as the fungi and bacteria it competes with, but faster and across a wider range of substrates. Some researchers believe the cockroach's enzyme repertoire, still only partially characterized, may eventually yield industrial applications in paper production, textile processing, and the breakdown of organic waste at scale. Historians of science sometimes note that the most useful chemistry is found not where we look first, but where we refuse to look at all. Leucophila maderae, the Madeira cockroach, produces fluorescence under ultraviolet light, a soft blue-green glow that emerges from the cuticle when illuminated at the right wavelength. This has been documented and replicated, but the reason for it remains genuinely unclear. Fluorescence in insects is not unique to this species. Certain beetles and scorpions also fluoresce, but in most cases researchers have proposed at least a plausible hypothesis. Mate recognition. UV reflection for thermoregulation, a byproduct of structural compounds. In the Madeira cockroach, no convincing functional explanation has yet been established. Scientists still debate whether the fluorescence is an active adaptation, something the cockroach evolved because it conferred some advantage, or a structural accident, a byproduct of the chemistry used to build the cuticle that happens to interact with UV light in a visually striking way. The cockroach itself lives in low-light environments and shows no behavioral response to UV illumination, which argues against the mate signaling hypothesis. Some researchers have proposed it may relate to the way the cuticle absorbs and re-emits solar radiation, but supporting evidence remains sparse. What is certain is this: under a UV lamp, a creature most people associate with dark corners and revulsion becomes something that glows with the quiet, cool light of deep ocean creatures. Perspective. It seems is partly a matter of lighting. Blabarus craniifer, known as the death's head cockroach for the skull-like marking on its pronotum, has developed one of the more remarkable defensive strategies in the cockroach family. It mimics the bumblebee. Not visually, the resemblance in shape is approximate at best, but chemically. The death's head cockroach produces cuticular hydrocarbons, whose chemical profile is sufficiently close to that of certain bumblebee species that predators which hunt by scent are reliably deceived. In field observations, lizards and birds that readily attack other cockroach species will hesitate back away or ignore the death's head cockroach entirely, not because they see a bee, but because they smell one, and the association between that scent profile and a painful sting is encoded in their experience. This is chemical mimicry, and it is considerably more sophisticated than the visual mimicry more familiar to naturalists. The cockroach does not need to look like a bumblebee. It only needs to smell like a threat. The marking on its back, that pale, skull-shaped pattern, is likely a coincidence of pigmentation, not a signal. Nature produced the name-worthy pattern and the genuinely useful adaptation in the same animal, but the pattern is theatre. The chemistry is the real protection. Macropenesthia rhinoceros, the giant burrowing cockroach of northern Australia, is the heaviest cockroach species on Earth. Adults reach 35 grams, roughly the weight of a golf ball, packed into an insect the size of a large. Is plum. Unlike the fast, skittish cockroaches most people picture, this species moves with the deliberate calm of an animal that has no serious predators in its native habitat. It spends most of its life underground in burrows. It excavates itself to depths of nearly a meter, using its heavily armoured front legs like miniature bulldozers. It does not fly. It does not scatter when disturbed. It simply waits, secure in the knowledge that its exoskeleton is thick enough to deflect most threats and its burrow deep enough to outlast most searches. What makes this species genuinely remarkable beyond its size is its lifespan under natural conditions. Giant burrowing cockroaches live for up to 10 years, an extraordinary duration for an insect. They raise their young inside the burrow for several months, feeding them on eucalyptus leaf litter that the parents drag underground and store. This combination of burrowing, food caching, extended parental care, and decade-long lifespan places this cockroach in behavioral territory more commonly associated with small mammals than insects. Australia characteristically produced a cockroach that behaves like a wombat. Most people are unaware that aquatic cockroaches exist, but several species in the genus Compsopelta and related groups spend significant portions of their lives in and around flowing water, and at least one Naucories is a confirmed semi-aquatic hunter. More striking is a species documented in cave streams in Southeast Asia that remains submerged for extended periods by trapping a bubble of air against its abdomen pressed beneath a water-repellent fringe of hairs along the body. Margin. This physical gill functions through the same principle as the diving bell spider's air sac. Oxygen diffuses into the bubble from the surrounding water as the insect consumes it, extending the functional breathable volume well beyond what the trapped air alone would provide. The cockroach in effect grows its own oxygen supply while sitting still. Researchers studying cave-adapted species have found that some populations show anatomical modifications, slightly flattened body profiles, more water-repellent cuticle chemistry, longer leg hairs, suggesting that aquatic life is not an accident but an evolutionary trajectory in progress. The cockroach family, which most people imagine as a fixed, uniform thing, is in fact still actively diversifying, exploring new habitats, testing new strategies, discovering new ways to persist in a world that has been trying to eliminate it for 300 million years. There is a story, possibly apocryphal, in which the biologist J.B.S. Haldane was asked what his study of nature had revealed about the mind of God. His answer, an inordinate fondness for beetles. Whether or not he said it exactly that way, the sentiment is biologically accurate. Beetles, order Coleoptera, comprise roughly 40% of all known insect species and approximately 25% of all described animal species on Earth. More than 400,000 species have been formally named and catalogued. Estimates of undescribed species run considerably higher. Some entomologists believe the true total may exceed 1 million. Beetles occupy every terrestrial and freshwater habitat on the planet, except the open ocean and the polar ice sheets. They eat everything: wood, dung, fungi, seeds, leaves, other insects, carrion, stored grain, living roots, and each other. They range in size from the featherwing beetles measured in fractions of a millimeter to the Titan beetle of South America, whose body alone exceeds 15 centimeters. No other group of animals has achieved this breadth of ecological occupation. When scientists want to understand how evolution produces diversity, how one body plan can expand to fill an entire planet's worth of ecological roles, they study beetles. The beetle is not one creature. It is evolution's most ambitious experiment still running. The defining feature of every beetle, the characteristic that unites all 400,000 species into a single order, is the elytra, a pair of hardened forewings that fold over the abdomen like a hinged shell, protecting the delicate hindwings and soft tissues beneath. The elytra are not used for flight. They are covers, protective cases, that allow the beetle to live in environments that would destroy the wings of any other flying insect. A beetle can burrow through soil, squeeze under bark, roll in dung, and drag itself through gravel, then open its elytra, unfold the membranous hindwings stored beneath, and fly away undamaged. The elytra are made of chitin, layered in precise orientations, a biological composite material whose structural properties vary across species depending on ecological role. In wood-boring beetles, they are dense and impact-resistant. In desert species, they are modified for water collection or heat management. In some aquatic beetles, they form an airtight seal that traps a breathing bubble. The same structure modified across 400,000 lineages solves 400,000 different problems. Engineers studying lightweight armor and foldable wing design have returned to beetle elytra repeatedly, not to improve on the design, but to understand it well enough to imitate it. Dynasties Hercules, the Hercules beetle of Central and South America, is frequently cited in discussions of proportional strength, and unlike many such claims in popular science, the underlying measurement is legitimate. Males of this species can carry loads equivalent to 850 times their own body weight. Scaled to human proportions, a rough analogy. Since the physics of scaling do not translate directly, this would correspond to a person lifting somewhere between 40 and 60 fully loaded city buses simultaneously. The comparison is imperfect, By design, insect muscle operates at a different mechanical scale than mammalian muscle, and small animals benefit from favorable surface area to volume ratios that make high proportional strength more achievable. The number is not meant to be taken literally, it is meant to be felt. The Hercules beetle uses this strength primarily for male competition. The long horn extending from its thorax is a wrestling implement, used to lever rival males off branches during fights over mating positions. The horn itself can equal or exceed the length of the beetle's body, making the animal look at full size like something a child imagined rather than something evolution produced. At up to 17 centimeters in total length, it is among the largest beetles by combined measurement on Earth. It is also, by any reasonable standard, absurd and magnificent. The bombardier beetle produces one of the most extraordinary defensive weapons in the animal kingdom through an entirely internal chemical reaction. Inside its abdomen, 2 separate chambers hold hydrogen peroxide and hydroquinone. When threatened, the beetle opens a valve between these chambers and introduces a third compound, a mix of catalytic enzymes, which triggers an explosive oxidation reaction. The resulting spray exits through a directional nozzle at the beetle's rear at a temperature approaching 100 degrees Celsius, the boiling point of water in a rapid pulsed burst that fires at a rate of roughly 500 pulses per second. Each pulse is a discrete micro-explosion. The spray is caustic, scalding, and accurately aimed. The beetle can rotate its abdomen to target threats from almost any angle, including directly forward. Frogs that attempt to swallow bombardier beetles have been documented spitting them back out, or in some cases the beetle survives, ingestion entirely firing inside the frog's stomach until the frog vomits. The internal reaction generates enough heat that it should, by rights, damage the beetle's own tissues, but the pulsed firing mechanism dissipates heat between bursts, preventing self-injury. Scientists studying this mechanism have proposed applications in fuel injection and aerosol delivery systems. The beetle solved controlled microcombustion roughly 200 million years before the internal combustion engine. Titanus giganteus, the titan beetle of the South American rainforest, holds the record for the largest beetle body in the world. By length, confirmed specimens reach 16 centimeters, roughly the span of an adult human hand from wrist to beak. Fingertip. Its mandibles are curved, serrated, and powered by a jaw muscle that occupies a disproportionate fraction of its thorax. Entomologists who have handled live specimens report that the mandibles can snap a wooden pencil cleanly in half and draw blood from a gloved hand without apparent effort. The beetle uses these jaws not for feeding adult titan beetles, do not eat at all, surviving entirely on fat reserves accumulated during their larval stage, but for defense against predators and possibly for combat with rivals, though this has rarely been directly observed in the wild. The larval stage, which may last several years inside dead hardwood trees, is when all actual feeding occurs. The larva itself is estimated to reach lengths of 30 centimeters, approximately the size of a human forearm, though no confirmed adult larva has ever been retrieved intact from the wild. Researchers still debate the precise duration of the larval stage, because no one has successfully tracked a titan beetle from egg to adult in uncontrolled conditions. Some things in nature remain stubbornly beyond the reach of careful observation. The great diving beetle Dytiscus marginalis is a fierce aquatic predator that hunts tadpoles, small fish, and aquatic invertebrates with a speed and aggression that seems improbable for an insect. What makes its underwater life possible is a simple but elegant solution to the problem of breathing before diving. The beetle surfaces and traps a bubble of air beneath its elytra, held against the abdomen by a fringe of hydrophobic hairs. This bubble functions first as a direct oxygen supply, and then as the oxygen is consumed as a physical gill. As the partial pressure of oxygen inside the bubble drops below that of the surrounding water, dissolved oxygen diffuses inward from the water, replenishing the supply. The beetle, by breathing slowly and staying still, can extend a single bubble's functional life well beyond its initial oxygen content. Like a scuba tank that refills itself from the surrounding ocean, the bubble shrinks as nitrogen escapes but continues delivering oxygen long after the original trapped air would have been exhausted. The diving beetle hunts with this system, feeds with it, and occasionally surfaces to refresh it, a brief visit to the air above, then back into the dim cold water below. It is in the most literal sense a creature that carries its own atmosphere. Fireflies are beetles family Lampyridae, and the light they produce is the result of one of the most chemically efficient reactions in biology. Inside a specialized organ in the abdomen, a compound called luciferin reacts with oxygen in the presence of an enzyme called luciferase, producing photons of light with almost no heat as a byproduct. Nearly 90% of the energy consumed by this reaction becomes visible light. Compare that to an incandescent bulb, which converts roughly 5% of its energy to light and wastes the rest as heat. And the firefly's cold light begins to look less like a curiosity and more like an engineering benchmark. Click beetles in the genus Pyrophorus produce a similar bioluminescence from two spots on their thorax, visible from a considerable distance in a dark forest. The chemistry is related but independently evolved, a case of convergent biochemical solutions to the same problem. Luciferase, extracted and synthesized, is now a standard tool in molecular biology. Researchers attach the gene that produces it to other genes they want to track, then simply watch for the glow to confirm the target gene has activated. The firefly's mating signal, refined over millions of years to attract a partner across a dark summer meadow, now lights the interior of cancer research laboratories. That particular journey of purpose from a field in summer to a laboratory bench is one biology did not plan but produced anyway. The jewel beetles of the family Buprestidae produce some of the most intense, saturated colors in the natural world. Deep, iridescent greens, electric blues, shimmering golds without using a single molecule of pigment. Their color is structural, produced entirely by the physical architecture of the cuticle, which is built in layers of precise thickness that cause light waves to interfere with one another. Certain wavelengths reinforce, others cancel, and the surviving wavelengths reach the eye as pure, saturated color. Because the color depends on the angle of observation, it shifts as the beetle moves. The same wing cover can appear green from one angle and copper from another, with no change in the underlying material. This principle, called thin film interference, is the same physics that produces rainbow patterns on soap bubbles and oil slicks. The difference is that the beetle's structure is precisely engineered nanometer-scale layers deposited with a consistency that a soap film, driven by surface tension alone, cannot achieve. Materials scientists studying jewel beetle cuticle have used it as a template for developing structural color in synthetic materials that do not fade, cannot be washed out, and require no dye chemistry. Jewel beetle wings were used as decorative inlay in jewelry and textiles for centuries across South and Southeast Asia, prized precisely because the color never dulled, long before anyone understood why. The Namib Desert receives less than 25 millimeters of rainfall per year, but it is regularly blanketed by fog rolling in from the Atlantic Ocean. Stenocara gracilipes, the Namib fog-basking beetle, has evolved to extract drinking water directly from that fog. Its back is covered in a pattern of small bumps, hydrophilic peaks, that attract and hold water droplets, surrounded by hydrophobic valleys that cause collected water to roll away toward the beetle's mouth. The beetle positions itself at the crest of a sand dune facing into the wind and tilts its body at a precise angle to maximize fog contact. Droplets collect on the bumps, grow until surface tension can no longer hold them, then slide down the hydrophobic channels directly to the beetle's head. The system requires no energy expenditure beyond the initial positioning. Engineers at MIT and several materials research institutions have used this structure as a direct template for water harvesting surfaces, coatings, applied to nets, panels, and building materials in water-scarce regions that passively collect moisture from air. Field trials in Chile, Morocco, and California have produced meaningful volumes of potable water from fog using beetle-inspired geometry. The Namib beetle did not invent fog collection. It perfected it across millions of years of the driest possible selective pressure, and we are only now catching up. The dung beetle Scarabaeus satirus, and this finding, published in a peer-reviewed study, remains one of the more quietly astonishing discoveries in insect navigation. can orient itself using the polarized light pattern of the Milky Way on moonless nights. Researchers tested this by observing dung beetles rolling their balls across open ground under different sky conditions, then fitting some beetles with small cardboard caps that blocked their view of the sky. Capped beetles wandered in irregular curves. Uncapped beetles rolled in straight lines. When the experiment was repeated inside a planetarium, with only the Milky Way projected overhead and all other light removed, the beetles maintained their orientation. They were not navigating by individual stars. They were using the diffuse light band of the galaxy itself as a compass, the only insect currently confirmed to do so. The precision required is remarkable. The Milky Way's light is faint, diffuse, and moves across the sky over the course of a night, yet the beetle tracks it well enough to maintain a straight course across open ground. Scientists still debate whether this ability is widespread among nocturnal beetles or specific to certain populations. What is not debated is that an animal the size of a thumbnail is using the galaxy to find its way home. That fact deserves a moment of stillness before moving past it. At the opposite extreme from the Titan beetle sits Pseudocella musawacensis, the smallest known beetle and one of the smallest free-living animals on Earth. Its body measures approximately 325 micrometers in length, about a third of a millimeter smaller than the period at the end of this sentence, as printed in a standard book. Its wings are not membranous surfaces but rows of individual bristles arranged in a feather-like pattern, hence the family name Ptiliidae from the Greek for feather. This wing structure is an adaptation to the physics of movement at extreme small scales, where air behaves more like a viscous fluid than a gas, and flat wing surfaces generate drag rather than lift. The feather wing moves through this thick, resistant air, the way an oar moves through water, each bristle slipping through individually, the gaps between them, allowing the stroke to be more efficient than a solid surface would permit. The beetle feeds on fungal spores and lives in the leaf litter and soil of forest floors worldwide, largely invisible to any observation that does not involve a microscope and considerable patience. It was not formally described until 2015, and its full biology remains poorly characterized. An entire animal active, mobile, feeding, reproducing, living in plain sight inside every forest on Earth, undiscovered by science until 11 years ago. The male stag beetle Lucanus cervus carries mandibles so enlarged they resemble the antlers of a small deer extending forward from the head in elaborate branched curves that can equal or exceed the length of the beetle's own body. These structures are entirely non-functional for feeding. The female stag beetle, whose mandibles are smaller and less ornate, can deliver a far more forceful bite. Her jaws are proportioned for actual use. The male's mandibles are built for one purpose: wrestling. Males compete for position on tree branches, particularly on the trunks of oak trees, where females gather to feed on sap by grappling with their mandibles, attempting to lift and throw rivals to the ground. The contests follow consistent rules and approach: a locking of mandibles, a test of leverage and body mass, and rarely result in serious injury. The winner remains on the branch. The loser falls, rights itself, and typically moves on. What is remarkable about this system is not the fighting but the cost. Growing and carrying these elaborate mandibles requires significant metabolic investment during the larval stage, which lasts 3 to 7 years underground. The male stag beetle spends the majority of its life preparing for an adult stage that lasts only a few weeks, in which its most important structure cannot be used for eating. It lives for the contest entirely. There is something in that which feels almost human. Stage 2 opens here with the first fact about intelligence, and it begins with a finding that challenged assumptions about what small-brained animals are capable of. When a group of cockroaches is placed in an environment containing multiple identical shelters, only some of which are large enough to comfortably house the entire group, the cockroaches will consistently and reliably select the appropriately sized shelter, not through any identifiable leader directing the group, but through a decentralized process that emerges from individual interactions. Each cockroach explores independently, leaving pheromone traces and making contact with others. When a shelter accumulates enough occupants to reach a chemical threshold, it begins attracting more individuals at an accelerating rate. Shelters that fill too quickly repel late arrivals through overcrowding signals. The system self-regulates toward the optimal choice without any individual cockroach having assessed all available options. Researchers at the Free University of Brussels modelled this process mathematically and found it matched the behaviour of certain human crowd dynamics, specifically the way groups of people making independent choices about where to gather tend to converge on the most comfortable space without explicit coordination. The cockroach did not develop democracy, but it did develop something that produces similar outcomes run entirely on chemistry without a single vote being cast. Cockroaches can be classically conditioned. This was demonstrated in controlled laboratory settings in which cockroaches were trained to associate a specific odour, peppermint, in one well-documented study with a sugar reward. After repeated pairings, the cockroach would extend its mouthparts in a feeding response when exposed to the peppermint odour alone with no food present. This is the same basic learning mechanism that Ivan Pavlov documented in dogs in the late 19th century stimulus association, and its presence in cockroaches was initially controversial. The cockroach brain contains approximately 1 million neurons. A human brain contains roughly 86 billion, and yet the cockroach demonstrates associative learning, extinction of learned responses when the reward is removed, and partial reacquisition when reward is reintroduced. Further experiments showed that cockroaches trained in the morning retain the learned association more reliably than those trained at night, suggesting the learning process is modulated by circadian rhythm, the same daily biological clock that governs memory consolidation in mammals. The capacity to learn, it turns out, does not scale simply with brain size. It scales with the precision of the circuits involved. The cockroach has had 300 million years to refine those circuits. What they lack in quantity, they have made up for in economy. Cockroaches do not simply memorize fixed routes through their environment. They maintain flexible spatial memory that updates in response to changing conditions. In laboratory maze experiments, cockroaches trained to navigate toward a food source through a specific pathway will spontaneously alter their chosen route when humidity or temperature along that pathway changes significantly. They are not following a rigid internal map. They are maintaining a map that includes environmental variables as active parameters and revising it when those parameters shift. This behavioral flexibility was documented by researchers in Japan who ran cockroaches through tea mazes under controlled humidity gradients over multiple sessions. Cockroaches that had established a reliable left-turn preference switched to the right-turn route when the left corridor was made significantly drier and switched back when conditions were restored. The memory was not erased by the switch. It was ranked. The cockroach appears to weight its stored routes by current desirability, not simply by familiarity. This kind of conditional spatial memory, preserving multiple route options and selecting among them based on present conditions, is a cognitive capacity that researchers more typically associate with vertebrates. The cockroach achieves it with a nervous system that weighs less than a grain of sand. Scale in biology is a poor predictor of sophistication. The cockroach nervous system does not concentrate control in a single central processor. Instead, it distributes decision-making authority across a series of ganglia discrete clusters of nerve tissue located in the thorax and abdomen, each one capable of coordinating the local muscles and sensory organs without consulting the brain. The thoracic ganglia control the legs independently. Researchers who have severed the connection between a cockroach's brain and its thorax have observed the legs continuing to produce coordinated walking movements in response to local stimulation. The abdominal ganglia manage the cerci, the reproductive organs, and the spiracle rhythms. The brain, located in the head, appears to function more as a modulator and integrator than a commander. It sets motivational state processes, complex sensory information, and influences the ganglia below, but it does not micromanage each step. This architecture has a practical consequence that robotics researchers find compelling. The system is fault-tolerant. Damage to one ganglion degrades local function without crashing the whole. Damage to the brain reduces behavioral flexibility without eliminating movement. The cockroach with a compromised brain is slower, less responsive, less adaptive, but it still walks, still breathes, still responds to threat. It is, in engineering terms, a system designed with graceful degradation built in from the start. Dung beetles placed in laboratory mazes have demonstrated a capacity for spatial problem solving that goes beyond simple trial and error. In a series of experiments where beetles were required to navigate toward a reward through a series of choice points, individuals that had previously been allowed to explore the maze passively without any reward present solved it significantly faster on subsequent rewarded trials than beetles with no prior exposure. This is latent learning, the acquisition of spatial information without immediate reinforcement stored and deployed later when it becomes useful. Latent learning has long been considered a marker of cognitive flexibility, the ability to gather information speculatively without a current need and retain it against future uncertainty. It has been documented in rats, birds, and primates. Finding it in beetles extended the list considerably. Some researchers remain cautious, noting that the experimental controls for simple habituation versus genuine spatial learning are difficult to make airtight at the scale of insect behavior. Scientists still debate whether what beetles demonstrate in maze studies constitutes true latent learning or a simpler form of environmental familiarization. What the debate itself reveals, however, is significant. The question is genuinely open. 40 years ago, it would not have been considered worth asking. During daylight hours, the dung beetle navigates using the sun tracking its position across the sky and compensating for its movement to maintain a straight rolling course away from the dung pile where competitors might steal its ball. This sun compass has been documented across multiple species and is considered well established. What is less expected is that the same orientation system appears to function at multiple scales on overcast days. Dung beetles use polarized light patterns in the sky, invisible to human eyes, to maintain direction. On partially cloudy days, they use wind direction as a supplementary cue, cross-referencing multiple signals simultaneously. The integration of these redundant navigational inputs, sun position, polarized light, wind, and on moonless nights, the Milky Way, gives the dung beetle a navigation system more robust than reliance on any single cue would allow. If one signal is unavailable, another takes precedence. The hierarchy of these cues and the conditions under which the beetle switches between them is still being mapped by researchers. What has emerged from this work is a picture of an animal whose navigational intelligence is not a single fixed ability, but a flexible layered system, the product of evolving in an environment where getting lost means losing everything you gather, worked to collect. A single bark beetle cannot kill a healthy tree. A healthy conifer's resin system, the sticky toxic sap it produces under bark, is sufficient to engulf and entomb an individual beetle attempting to bore in. What bark beetles have evolved instead is a coordinated mass attack strategy that overwhelms this defense through sheer simultaneous pressure. When a pioneer beetle locates a suitable tree, it releases aggregation pheromones that recruit additional individuals from the surrounding area. As more beetles arrive and begin boring, they add their own pheromone signals, amplifying the recruitment call in a positive feedback loop. Within hours, hundreds or thousands of beetles may be boring simultaneously across the entire surface of a tree, stimulating resin production faster than the tree can sustain it. The resin system exhausts itself. The tree, now unable to defend any single entry point, is overwhelmed. This coordinated attack is not planned. No individual beetle assesses the tree's defenses or calculates the number of recruits needed. The coordination emerges from the chemical cascade itself, each beetle responding to the same signal, the collective behavior arising from individual responses to shared chemistry. It is, in ecological terms, a highly effective strategy. In forests, weakened by drought or warming temperatures, conditions that reduce resin production, the threshold for successful mass attack drops dramatically, which is why bark beetle outbreaks have expanded significantly across North American and European forests in recent decades. Wasmannia rove beetles and several species across the family Staphylinidae have evolved the ability to infiltrate ant colonies by producing cuticular hydrocarbons that mimic the chemical signature of the host ant species. Ants recognize nestmates almost entirely through chemical contact. The colony's identity is encoded in a blend of hydrocarbons on the cuticle surface, and individuals that match the colony's blend are accepted. Those that do not are attacked. The rove beetle produces a near-perfect chemical forgery of this blend, allowing it to move freely through the colony, be groomed by workers, fed by trophallaxis, and even gain access to the brood chamber where it feeds on ant larvae. The deception is not static. Studies have shown that as ant colonies adjust their chemical signatures over time, which they do in response to environmental changes and colony composition, some rove beetle populations track this shift, updating their own chemical profile to maintain the match. This is not learned behavior in a conscious sense. It is an evolutionary arms race, compressed into individual lifetimes, the beetle's chemistry co-evolving with the colony's identity system in real time. You could spend a lifetime studying ant colonies and never suspect that some of what you were watching was not an ant. Microphorus burying beetles, also called sexton beetles, display a level of cooperative parental investment that is among insects almost without parallel. When a male and female locate a small vertebrate carcass, a dead mouse or sparrow, they work together to bury it, stripping away fur or feathers, rolling the body into a compact ball, and covering it with soil in a process that can take several hours of sustained, coordinated effort. Once buried, they excavate a chamber around it, treat the carcass surface with oral and anal secretions that slow bacterial decomposition, and hollow out a small cavity at the top into which the female lays her eggs. When the larvae hatch, both parents remain to feed them, regurgitating pre-digested carcass material directly into the larvae's mouths in a behavior that resembles structurally the chick-feeding behavior of birds. Parents also regulate the brood size by consuming excess eggs if the carcass is too small to support the full clutch, a form of resource matching that requires assessing carcass mass relative to offspring number. The male typically departs first. The female continues care until the larvae pupate. No other beetle family approaches this complexity of coordinated parental behavior. In the insect world, Microphorus is quietly remarkable, a creature that buries its dead and feeds its young simultaneously with more care than most people would expect from something with 6 legs. The mechanism by which burying beetles match their brood size to available resources is not guesswork. Female Microphorus beetles regulate egg production and selectively cull larvae with a precision that implies genuine assessment of carcass mass. Experiments in which researchers provided beetles with carcasses of controlled, varying sizes found that females laid more eggs on larger carcasses and fewer on smaller ones, and that when carcasses were switched after egg laying, females adjusted the surviving brood through selective infanticide to match the new resource level. This adjustment happens before the larvae have consumed any significant portion of the carcass, which means the female is projecting forward, estimating how many offspring the resource can support and calibrating her investment accordingly. The sensory mechanism is not fully understood. Researchers believe olfactory cues from the carcass, volatile compounds whose concentration reflects mass and decomposition stage, provide the primary signal, but exactly how the beetle converts a smell into a numerical brood decision remains an open question. What is clear is that the decision is made, it is made accurately, and it is revised when conditions change. A beetle that smells a dead mouse calculates the arithmetic of survival and adjusts the number of her children accordingly, is doing something that feels, despite every instinct to resist the word, like planning. The bombardier beetle does not rely solely on its chemical cannon. Before deploying that weapon, it employs a prior strategy that appears in high-speed video analysis to be deliberate. It goes entirely still. When first contacted by a predator, a frog's tongue, a bird's beak, a spider's foreleg, the bombardier beetle ceases all movement, becoming limp and unresponsive. Many predators, conditioned to pursue moving prey momentarily, relax their grip or reduce pressure on a motionless target. That relaxation creates a gap. In that gap, the beetle fires. The sequence stillness, then explosion, is more effective than either strategy alone. A beetle that fires immediately may be swallowed before the spray reaches the predator's sensitive tissues. A beetle that only plays dead remains vulnerable once the predator resumes investigation. Together, the two behaviors create a timed trap. The first stage buys the physical space the second stage requires. Researchers studying this sequence noted that the delay between contact and firing was remarkably consistent across individuals, approximately 200 milliseconds, suggesting a fixed neural circuit rather than a learned decision. The beetle does not calculate the optimal moment to fire. Its nervous system was built with the timer already set. Evolution, in this case, did the thinking in advance. In 2015, researchers at the Free University of Brussels published a study demonstrating that individual cockroaches within the same population show consistent, repeatable differences in behavior that qualify under the scientific definition as personality. Specifically, they documented variation along a boldness-shyness axis. Some individuals consistently explored new environments quickly, spent more time in open spaces, and initiated contact with novel objects. Others consistently remained sheltered, moved cautiously, and retreated from unfamiliar stimuli. These differences were stable across time and across different testing situations. The same individuals that were bold in one context were bold in another weeks later. Personality in behavioral ecology is defined as consistent individual variation in behavior that cannot be explained by differences in current state, age, or size alone. By that definition, the cockroaches had it. The study also found that groups composed of mixed personality types made better collective decisions, reached shelter faster, and wasted less time in consensus than groups composed entirely of bold or entirely of shy individuals. Diversity in temperament, in other words, produced better group outcomes than uniformity. That finding appeared in a paper about cockroaches. It has implications that extend considerably further than entomology. When a cockroach enters an unfamiliar space, it does not wander randomly. It moves to the nearest wall and follows it. This behavior, called thigmotaxis from the Greek for touch and arrangement, reflects a deep preference for maintaining contact with a solid surface on at least one side of the body. The wall provides sensory grounding. The cockroach's body hairs and leg mechanoreceptors register the surface continuously, giving it positional information about its environment, even in complete darkness. Following the wall also guarantees systematic coverage. A cockroach tracing the perimeter of a room will eventually encounter every exit, every gap, every food source along that boundary without needing to maintain a mental map of the interior. It is a search algorithm built into the body. Computer scientists studying coverage algorithms, the problem of how a robot or agent can systematically explore an unknown space, have noted that wall following is one of the simplest and most reliable solutions, provided the space has no isolated interior obstacles. The cockroach arrived at this solution not through reasoning, but through millions of years of selection in environments where gaps in walls meant exits and exits meant safety. The strategy works in sewers, in caves, in the walls of buildings, and in the interiors of spacecraft where, as it happens, cockroaches have also travelled. In the Namib and Sahara desert's midday ground, temperatures on open sand can exceed 70 degrees Celsius, hot enough to cook an egg, and far beyond what a beetle's leg tissue can tolerate for more than a few seconds. Dung beetles crossing open ground during the hottest part of the day face a genuine thermal emergency with every step. Researchers, observing this behavior, documented something unexpected. Beetles that had been rolling dung balls would periodically stop to climb to the top of their ball and stand there, elevated a few centimeters above the scorching sand, motionless for several seconds. Then they would descend and continue rolling. Thermal imaging confirmed that the dung ball surface, being cooler than the sand below, provided measurable relief, and that the elevation brought the beetle into slightly cooler air. The climbing behavior occurred at temperature thresholds, not randomly, and ceased when the ground cooled in the afternoon. Whether this constitutes tool use in the strict sense defined as using an external object to achieve a goal is something researchers still debate. The beetle does not carry the ball for this purpose. It uses it for this purpose when the need arises. The distinction is philosophically interesting and practically small. The beetle climbs when it is too hot. It descends when it is not. That is, at minimum, situational problem-solving with available resources. The tiger beetle family Cicindelidae is among the fastest-running insects on Earth, capable of covering ground at approximately 125 body lengths per second. At this speed, a genuine perceptual problem emerges. The beetle's eyes cannot process visual information fast enough to track its target in real time. The image on its retina blurs into uselessness. The tiger beetle at full sprint is functionally blind. It responds to this limitation by stopping reacquiring its target visually during the stationary moment, then accelerating again, producing the characteristic stop-start pursuit pattern that observers have noted for decades. Without understanding the cause, high-speed filming and analysis of eye movement confirmed the visual processing bottleneck. The beetle is not hesitating, it is recalibrating. What makes this more remarkable is what happens during the blind sprint phase. The beetle does not run randomly. It runs in a straight line toward the last known position of its prey, adjusted by a predictive calculation, essentially where the prey will be by the time vision is restored, assuming it continues at its current speed and direction. This predictive dead reckoning occurs automatically encoded in the neural circuitry that initiates the sprint. The tiger beetle does not think about where its prey will be. Its nervous system thinks for it in the fraction of a second before the legs begin to move. The collective shelter selection behavior of cockroaches, first introduced in Fact 31, operates through a mechanism that researchers have characterized with some precision as a form of quorum sensing. Each cockroach that enters a shelter deposits aggregation pheromone. Other cockroaches detect this pheromone and are drawn toward the source. As the concentration of pheromone in a shelter rises, it attracts individuals at an accelerating rate, a positive feedback loop. But the system has a natural ceiling. As crowding increases beyond a comfortable density, physical contact, signals, the tactile experience of being jostled, begin to counteract the chemical attraction. The result is a self-limiting aggregation. Shelters fill to comfortable capacity and then stop attracting more individuals, while less used shelters continue broadcasting their pheromone signal to latecomers. The elegance of this system is that it requires no individual to assess overall population or shelter capacity. Each cockroach responds only to local chemical and tactile signals. The group-level outcome optimal distribution across available shelters emerges from those individual responses without coordination. Biologists refer to this as stigmergy collective intelligence, built not from shared planning but from shared responses to a shared environment. It has been found in ants, termites, and slime molds. Finding it in cockroaches expanded the list of animals capable of producing order without anyone being in charge. Cockroaches display one trial learning for aversive events. They can update their spatial behavior based on a single negative experience without repeated exposure. In controlled experiments, cockroaches that received a mild electric shock in a specific location of a familiar arena subsequently avoided that location during free movement even when no shock was present, and even when the arena's lighting and odor cues were altered to reduce the salience of the specific spot, the avoidance persisted for days. This single trial aversive learning is ecologically significant. In a natural environment, an encounter with a predator, a toxic substance, or a physical trap rarely offers a second chance for learning. An animal that requires multiple exposures to update its behavior is an animal that may not survive the curriculum. The cockroach's nervous system appears to be structured for rapid aversive. Updating a single strong negative event writes a durable avoidance memory that resists extinction under normal conditions. Researchers found that this memory was consolidated most effectively when the aversive event occurred during the cockroach's active phase, the nighttime hours, which aligns with the circadian modulation of learning observed in classical conditioning studies. The cockroach remembers bad nights particularly well, in that it is perhaps not so different from any other animal that has learned the hard way to be careful. Whirligig beetles, family Gyrinnidae, live on the surface film of still and slow-moving water, and they navigate this two-dimensional world using a sensory system unlike anything else in the insect order. Their antennae rest on the water surface and detect the ripple patterns generated by objects in their vicinity, their own movement, the movement of prey, and the reflections of ripples bouncing back from nearby obstacles like stones or reeds. By analyzing the timing and amplitude of returning ripple echoes, the whirligig beetle constructs a real-time map of its immediate surroundings. This is surface echolocation, functionally analogous to the sonar used by bats and dolphins, but operating through water surface waves rather than sound waves in air. The beetle's antennae are divided into two functional sections. The upper segment detects airborne vibrations, and the lower segment resting on the meniscus detects surface waves. The two systems operate simultaneously, giving the beetle information from both the air above and the water below. Whirligig beetles also have divided eyes. Each eye is split into an upper half that sees above the waterline and a lower half that sees below it, giving them simultaneous vision in two optical environments. They are in almost every sensory dimension animals built to inhabit a boundary. Not fully in the water, not fully out, perfectly suited to the line between. When a cockroach becomes infected with a pathogen, it does not simply continue its normal routine while the infection runs its course. Its behavior changes in ways that measurably reduce the risk of transmission to other members of its group. Infected individuals spend more time in the periphery of aggregations rather than the center, reducing their contact rate with healthy nestmates. They groom themselves more frequently, a behavior that reduces the surface load of transmissible spores or bacteria. In some documented cases, infected individuals leave the aggregation shelter entirely and remain isolated until either recovery or death. Researchers studying this phenomenon have ruled out the possibility that the behavioral changes are simply a byproduct of illness-induced weakness. The spatial repositioning and increased self-grooming appear to occur before the cockroach shows visible signs of physical debilitation, suggesting an active early-stage response rather than a passive consequence of declining health. Whether the cockroach is in any meaningful sense aware that it is protecting others is a question that cannot currently be answered. What can be measured is the outcome groups containing infected individuals that display this behavioral shift show lower secondary infection rates than groups in which infected individuals maintain normal social contact. The cockroach does not understand epidemiology. Its behavior produces epidemiological outcomes regardless. Inside the pupa of a metamorphosing beetle, something occurs that remains one of the more genuinely strange processes in developmental biology. The larval body does not simply rearrange itself into the adult form. In many beetle species, a significant portion of the larval tissue breaks down entirely, dissolving into an undifferentiated cellular soup before being reconstructed into adult structures by clusters of cells called imaginal discs, which were set aside in the larval body from the very beginning and held dormant throughout the feeding stage. These imaginal discs carry encoded in their developmental programming the blueprint for every adult structure: the elytra, the adult legs, the compound eyes, the reproductive organs. The hormone ecdysone triggers their activation, and juvenile hormone declining at the correct moment releases the brake that had kept them dormant. The timing of this hormonal cascade determines not just whether metamorphosis occurs, but what kind of adult emerges: body size, reproductive readiness, wing development, all modulated by the hormonal environment the larva experienced during its feeding stage. A larva that fed well and experienced optimal temperatures produces a larger, more reproductively capable adult. The adult beetle's quality is written into it not at the moment of pupation but across the entire duration of its larval life. The beetle becomes what its larva experienced. That principle stated plainly applies to more than beetles. Cockroach societies, if that word can be applied to groups of animals whose social bonds are chemical rather than cognitive, are held together primarily by aggregation, pheromones, volatile, and contact compounds produced in the hindgut, and deposited in feces which attract other cockroaches and encourage them to remain in proximity. These pheromones are not simple attractants. They carry information. The chemical composition of aggregation pheromone varies with the age, sex, and reproductive state of the individual producing it, meaning that a cockroach approaching a pheromone-marked surface can extract from the blend a rough census of who has been there and in what condition. Nymphs, juvenile cockroaches, are particularly sensitive to aggregation pheromones produced by adults of their own species, and this sensitivity appears to serve an important developmental function. Nymphs raised in environments with adult pheromone develop more normally, reach reproductive maturity faster, and show better survival rates than nymphs raised in chemical isolation. The pheromone is not just a signal to gather. It is a developmental cue, a chemical message that tells the young animal it is in the right place among its own kind, in conditions where survival is probable. Community for the cockroach is not a social choice, it is a chemistry, and that chemistry shapes who the animal becomes. Most people do not know that cockroaches produce sound. The mechanism, stridulation, the rubbing of specialized body surfaces against one another to generate vibration, is more commonly associated with crickets and cicadas, but it is present in a significant number of cockroach species, particularly those that live in more structured social environments. In stridulating cockroach species, the sound-producing structures are typically located on the abdomen or the edges of the thorax, and the sounds produced range from barely audible to humans to frequencies entirely outside our hearing range. The function of these sounds varies by species and context. In some, stridulation appears during male-male competition, producing a warning or threat display without physical contact. In others, it occurs during courtship with the rhythm and amplitude of the sound apparently conveying information about the male's condition. In a few species, stridulation has been documented in nymphs disturbed by predators, a distress signal that may trigger defensive responses in nearby adults. The sounds cockroaches make are not random vibrations produced by incidental movement. They are structured, context-dependent signals, a form of acoustic communication operating just below the threshold of human attention in the walls and dark spaces where these animals have always lived, speaking to each other in a language no one thought to listen for. The Madagascar hissing cockroach Gromphodorhina portentosa resolves most of its male dominance contests through sound rather than physical combat. When two males encounter each other, they rear up, make body contact, and begin producing their characteristic hiss, a forced expulsion of air through the abdominal spiracles that creates a sustained, audible sound unlike anything else in the cockroach family. The hiss serves as a threat. Display males assess each other's hiss rate, amplitude, and duration before committing to physical engagement. In many cases, the contest ends at this stage. The smaller or less vigorous male hisses less convincingly, and the dominant male's response escalates in intensity until the subordinate withdraws. Physical fighting occurs, but is less common than vocal standoff. Researchers studying the social hierarchies of hissing cockroach colonies found that dominant males hiss more frequently at higher amplitude and maintain more consistent hiss rhythms than subordinate males, and that these acoustic 24 differences are detectable by other males, who respond differentially to dominant versus subordinate hiss profiles. The hissing cockroach, in other words, negotiates. It uses sound to convey size, condition, and willingness to escalate information that, if accurate, allows both parties to avoid a costly fight. Diplomacy conducted through spiracles in the dark. What distinguishes the Madagascar hissing cockroach's sound production from that of other stridulating insects is its mechanism. Crickets and katydids rub body parts together. The hissing cockroach uses its respiratory system directly, forcing air through modified spiracles on its abdomen in a controlled, forceful expulsion that produces the distinctive hiss. This is functionally analogous to the way a human produces sound through the larynx. Exhaled air passing through a constricted opening generates vibration. The cockroach has no larynx, no vocal cords, and no mouth cavity that amplifies sound. Its voice is its breath, shaped by the opening and closing of spiracles that evolved for gas exchange and were repurposed, or perhaps co-opted, is the better word for communication. The hissing cockroach is one of the largest cockroach species, reaching 7 to 8 centimeters in length, and its spiracles are correspondingly large enough to produce a sound audible to humans from several meters away. In its native Madagascar, it lives in forest floor, leaf litter, and rotting logs, where visual signals are largely useless in the darkness, and chemical signals may not travel fast enough to resolve a territorial dispute quickly. Sound in that environment is fast, directional, and honest, difficult to fake at full amplitude without the physical capacity to back it up. In cockroach species that engage in active mate choice, females do not respond equally to all males. Studies of several species, including the German cockroach and various tropical forest species, have documented that females spend significantly more time in contact with males, whose cuticular pheromone profiles are more chemically complex, containing a greater variety of hydrocarbon compounds in more precisely balanced ratios. The preference for complexity appears to function as an honest signal. Producing a diverse, precisely blended pheromone profile requires good metabolic health, adequate nutrition during development, and genetic diversity at the loci that regulate hydrocarbon synthesis. A male in poor condition produces a simpler, less balanced blend. A male in peak condition produces the full, elaborate profile. The female, by preferring complexity, is selecting without any conscious awareness of the underlying biology for the indicators that best predict offspring quality. This is the same principle that underlies the peacock's tail, the nightingale's song, and the bowerbird's elaborate constructed display. The medium is different in each case. The logic is identical. Cockroach courtship, conducted in darkness through chemical signals no human nose can detect, follows the same evolutionary grammar as every other system of mate choice that biologists have studied. Because the problem being solved is always the same: how to find, from among the available options, the best possible future for your offspring. Trophallaxis, the direct transfer of liquid food between individuals through mouth contact, is well documented in ants and bees, where it functions as both a nutritional distribution system and a means of spreading colony-wide chemical information. It is less widely known that cockroaches practice a form of trophallaxis as well, particularly between adults and nymphs in species that live in stable aggregations. An adult cockroach that has fed will, when contacted by a hungry nymph, regurgitate a small quantity of crop contents, partially digested food, which the nymph consumes directly. This transfer passes not only nutrients, but gut microbiome components. The bacteria and protozoa that populate the cockroach's digestive system, which nymphs require for proper digestion of complex plant material, but cannot acquire from the environment alone. A nymph, raised in complete isolation from adults, develops an impoverished gut microbiome and significantly reduced digestive efficiency. The mouth-to-mouth transfer is in this sense more than feeding. It is inoculation the older generation, seeding the younger with the biological tools they will need to survive. The cockroach does not teach its young in any way we would recognize as teaching, but it passes something essential forward in the oldest possible way through contact and proximity and the willingness to share what it carries inside. Young cockroaches do not discover their foraging routes independently. When nymphs first begin moving beyond the shelter aggregation, they follow chemical trails deposited by adults, contact pheromones left in fecal deposits, and tarsal secretions along routes that experienced individuals have already established as productive. The nymph, moving along these chemical pathways, arrives at food sources without having searched for them, and in doing so begins building its own spatial memory of productive routes before it is capable of independent exploration. Researchers tracking nymph movement in controlled arenas found that nymphs raised with access to adult chemical trails established reliable foraging routes significantly faster than nymphs raised in chemically neutral environments, showed better body condition at the end of the experimental period. The trail system functions as a form of cultural transmission, not culture in the human sense, with conscious teaching or deliberate demonstration, but culture in the biological sense, information accumulated by one generation, encoded in the environment and acquired by the next, without direct instruction. The adult cockroach does not guide the nymph. It leaves traces of where it has been, and the nymph follows. The knowledge travels forward not through memory but through chemistry written on the floor of the world the nymph is learning to navigate. Bark beetles communicate with extraordinary chemical specificity. The aggregation pheromones that recruit individuals to a suitable host tree are not generic attractants, they are species-specific blends of volatile compounds whose precise ratios function as molecular passwords recognized by members of the producing species and largely ignored by others. A single compound present in slightly the wrong concentration renders the blend unrecognizable. This specificity serves an important ecological function. Dozens of bark beetle species may inhabit the same forest, targeting overlapping host tree species, and the chemical password system allows each species to recruit its own kind without accidentally attracting competitors. Some species go further. They produce anti-aggregation pheromones, compounds that once the tree is fully colonized suppress further recruitment and direct latecomers to adjacent trees. The same chemical communication system that assembles the mass attack disassembles it at precisely the right moment, preventing overcrowding that would reduce individual reproductive success. The sophistication of this two-phase signaling recruit then disperse is difficult to improve upon from an engineering standpoint. Pest management researchers studying bark beetle outbreaks have attempted to exploit these pheromone systems for control purposes, deploying synthetic attractants to lure beetles into traps. The beetles, after initial attraction, often develop behavioral resistance, adjusting their response thresholds in ways that reduce trap efficacy over time. The password, it seems, can be changed. Male dung beetles compete for mates partly through a behavior that functions as a physical fitness test, the construction and rolling of a dung ball. A male that can shape a large spherical ball from fresh dung and roll it in a straight line away from the dung pile. Navigating competition theft attempts from rival males and uneven terrain demonstrates through that single act a constellation of qualities relevant to reproductive fitness. Ball rolling requires physical strength, coordination, navigational ability, and the capacity to defend a resource against competitors while simultaneously moving it. Females observe multiple males and preferentially associate with those rolling, larger, more symmetrical balls along straighter trajectories. Some females ride on top of the ball as the male rolls it, a behavior that appears to serve an assessment function, allowing the female to evaluate the male's rolling ability directly, while contributing additional weight as a test of his strength. The ball itself, once the mating pair buries it, becomes the brood mass in which eggs are laid and larvae develop, meaning the female's assessment of ball quality directly correlates with the nutritional resource her offspring will receive. The male's courtship display and the offspring's food supply are the same object. The performance is the provision. What looks like a dance is simultaneously an audition and a grocery run. Burying beetles communicate with their larvae through a mechanism that is in its intimacy genuinely unexpected. They stridulate, produce vibrations by rubbing body parts together, and transmit those vibrations through the carcass on which the larvae are feeding. The larvae detect these vibrations through mechanoreceptors in their legs and respond by moving toward the vibrating adult, where they receive regurgitated food. The signal is not simply a dinner bell. Researchers have found that the vibration pattern produced by parents stridulating on the carcass differs from vibrations produced in other contexts, and that larvae respond specifically to the parental signal rather than to mechanical, the disturbance of the carcass in general. The carcass in this system functions as a communication medium, a substrate that transmits parental signals to offspring in the darkness of the underground burial chamber, where light-based signaling is impossible and airborne chemical signals may be too diffuse to reliably locate. The parent touches the dead, and the vibration travels through it to the living. There is something in that image that resists easy categorization. It is not sentiment exactly. It is simply a fact that arrives with more weight than most facts do. A small beetle underground, in the dark, pressing its body against a carcass to call its children to dinner through the only medium available. North America alone hosts more than 150 firefly species, and the majority of them communicate through bioluminescent flash patterns that are species-specific, each species producing a characteristic sequence of flashes, pauses, and flight patterns that allows individuals to identify potential mates of their own kind in a field shared by multiple species simultaneously. The flash pattern encodes species identity in its timing. The duration of each flash, the interval between flashes, the number of flashes per sequence, and the flight trajectory during flashing are all parameters that vary consistently between species and remain stable within them. Males typically flash while flying. Females respond from perches below. The male's flash advertises his species, his location, and in some species aspects of his condition, since males in better physical health produce brighter, more precisely timed flashes. Females assess the pattern and respond with a species-matched answering flash at a precise delay interval. The delay itself is part of the recognition system. A male receiving a response at the correct interval approaches. A response at the wrong interval is ignored. This temporal precision, measured in tenths of a second, is maintained consistently enough that researchers can identify firefly species in the field using only stopwatch timing without capturing a single specimen. The flash is a name, a location, a resume, and a question. The answering flash is a yes. Female fireflies of the genus Photuris have evolved one of the more cold-blooded strategies in the insect world. They mimic the answering flash signals of females from other firefly genera, particularly Photinus, to lure the males of Photinus. Those species within striking distance then capture and eat them. These females, called femmes fatales in the entomological literature, can produce the correct answering delay for multiple Photinus species switching between mimicked signals as different male species fly past. The deception is precise enough to be effective. Photinus males approach and land expecting to find a receptive female of their own species and instead find a predator. The femmes fatales benefit nutritionally from this deception. The males they consume contain defensive steroidal compounds called lucibufagins that Photuris females cannot synthesize themselves but incorporate into their own tissues, making them toxic to predators like spiders. By eating Photinus males, the female acquires chemical defenses she could not otherwise produce. The signal she mimics is thus doubly valuable. It provides food, and that food provides protection. The firefly flash system, built over millions of years as a private language between mates of the same species, was infiltrated character by character by a predator patient enough to learn the code. Communication, wherever it exists in nature, creates the possibility of its own exploitation. Many beetle species produce sound when handled or threatened, a sudden sustained stridulation generated by rubbing the abdomen against the inner surface of the elytra, or by rubbing specialized files on the thorax against ridged surfaces on adjacent body segments. The sound produced is not loud by mammalian standards, but it is precisely calibrated to startle. It occurs at a frequency and amplitude that consistently triggers a flinch response in the small vertebrate predators, lizards, rodents, shrews, that are most likely to attempt consuming a beetle. The startled predator momentarily releases or relaxes its grip, and the beetle uses that fraction of a second to deploy secondary defenses, drop from a height, or simply run. In some species, the stridulation continues throughout the escape attempt. A sustained alarm that may also recruit conspecifics or alert other prey animals in the vicinity that a predator is active nearby. Researchers studying predator-prey interactions have found that naive predators exposed to stridulating beetles for the first time show significantly more hesitation and release behavior than those exposed to silent prey of equivalent size. The sound is not painful. It is not a warning of toxicity. It is a startle, a sudden intrusion into a moment of confident predation. The beetle does not fight back. It interrupts. Cockroaches can distinguish between related and unrelated individuals using the chemical profiles of their cuticles, the blend of hydrocarbons on the body surface that functions simultaneously as waterproofing, disease protection, identity signal. In experimental settings where cockroaches were given the choice between sheltering with siblings versus unrelated individuals of the same species, they showed a measurable preference for siblings spending more time in shelters containing familiar kin-related chemical profiles. This kin recognition operates through contact chemoreception. The cockroach tastes through its leg, chemoreceptors, and antennae, the hydrocarbon blend of individuals it encounters, and compares it to an internal reference template. That template appears to be calibrated through early life experience. Nymphs raised together develop shared or overlapping chemical profiles and recognize each other as familiar, even if genetic relatedness is subsequently disrupted. The system is not infallible. It can be fooled by shared environment, which produces chemical convergence even between unrelated individuals. But under natural conditions, where related individuals typically share both genetics and early environment. The chemical recognition system reliably produces kin preference. A cockroach prefers the company of its family. It cannot say why. It knows through its feet and through the fine-tuned chemistry of a body that has been refining this recognition for longer than most family trees extend. When food and shelter become scarce, cockroach aggregations do not simply continue growing until collapse. The chemical signals that normally hold the group together shift in composition, and the aggregation begins to dissolve at its edges. Individuals at the periphery receive weaker pheromone signals and experience more competition for resources, and they disperse. In some species, this dispersal is accompanied by a documented increase in cannibalism. Adults consuming eggs and early instar nymphs, selectively removing the most energetically costly members of the group. This cannibalism is not indiscriminate. Studies have found that adults preferentially consume unrelated eggs over their own, and that the rate of cannibalism scales with the severity of resource limitation rather than occurring as a fixed behavior. The group does not decide to reduce its size. The chemistry of scarcity produces behavioral changes in individuals, and those individual changes produce a population-level response. The aggregation contracts to a sustainable core. The dispersed individuals either find new resources or perish. What remains is a group calibrated, however roughly, to what the local environment can support. Ecologists studying population dynamics have noted that cockroach populations rarely crash catastrophically in stable environments. They oscillate, contract, and expand in response to resource availability with a responsiveness that more complex animal societies sometimes fail to achieve. Necrophorus burying beetles, reintroduced here from their earlier appearance in stage 2, but now in a different light, have been observed by naturalists for centuries, with a mixture of fascination and discomfort that the animals themselves seem entirely indifferent to. They locate the dead through extraordinary olfactory sensitivity. They can detect the volatile compounds of early decomposition from distances exceeding a kilometre, navigating to a carcass within hours of death, often arriving before any other large scavenger. Once there, they assess it, prepare it, and inter it, a process that involves stripping, compressing, treating, and burying the body with a thoroughness that no other insect approaches. The ancient Egyptians, who encountered large Necrophorus species along the Nile, observed their behavior without knowing its mechanics, and understood intuitively that these beetles were associated with death and transformation. They did not worship the burying beetle the way they worshipped the scarab, but they noted its presence at the threshold between life and decomposition, which is precisely where it operates. In a forest ecosystem, the burying beetle is the first and most efficient processor of small vertebrate death, returning nutrients to the soil, reducing pathogen load in decomposing tissue, and completing a cycle that the living depend upon. Without ever witnessing, it attends to the dead so that the living may continue. Ground beetles family, Carabidae, one of the largest beetle families with over 4,000 species, deposit chemical markers from specialized glands as they move through their home range. These secretions adhere to the substrate and degrade at a rate that encodes temporal information. A fresh trail signals recent occupation. A faded trail signals absence. Other beetles encountering these trails can, through contact chemoreception, assess not just that another individual has been present, but approximately when distinguishing a trail laid minutes ago from one laid hours ago by the intensity and completeness of the chemical signal. This time-stamped territorial marking allows ground beetles to maintain spatial awareness of competitors' movements without direct contact, reducing the energy cost of territorial defense while still communicating occupancy reliably. Some carabid species have been found to produce trail chemicals whose degradation rate is precisely matched to their own patrol cycle. A fresh trail from their last circuit is detectable by competitors, but faded enough by the next patrol that the beetle itself does not follow its own old signal. The chemistry self-erases on a schedule tuned to the beetle's behavior. Whether this precision evolved deliberately or emerged as a byproduct of the general chemistry involved is something scientists still debate, but the functional result is a territorial system that updates itself automatically with every passing hour. Inside the bark of a colonized tree, bark beetles construct gallery systems whose architecture reflects a division of labor as consistent as anything found in social insects. In most bark beetle species, it is the female who excavates the primary egg gallery, a tunnel running parallel to the wood grain, with precisely spaced egg niches cut into the walls at regular intervals. The male typically remains near the gallery entrance, clearing frass the sawdust-like excavation debris, and blocking the tunnel against parasites, predators, and competing beetles attempting to enter. As the female excavates forward, the male works backward, maintaining the gallery behind her. The larvae that hatch from the eggs excavate their own perpendicular tunnels outward from the maternal gallery, following the wood grain in directions that minimize competition with siblings, producing the characteristic engraved pattern, visible on debarked wood, that has been described by those who find it beautiful as looking like ancient script. Each species produces a characteristic gallery pattern so consistent that entomologists can identify the beetle from the engraving alone without seeing the insect. The gallery is the beetle's signature. It is also its legacy, remaining readable in the wood for years after the beetles have moved on, a record of labor and care pressed into a dying tree. In stable cockroach aggregations, individuals that show signs of illness or injury receive measurably different treatment from healthy nestmates. Not avoidance, as one might expect from social immunity concerns, but increased grooming. Healthy cockroaches in the same aggregation spend more time grooming ill individuals than healthy ones, concentrating their antennal contact and oral grooming behavior on the affected individual's body surface. Researchers documenting this behavior initially hypothesized it might represent pathogen harvesting healthy individuals, consuming infectious material for nutritional reasons. But subsequent studies found that the grooming behavior increased, even in cases where the illness was non-transmissible, arguing against a purely self-interested interpretation. The most current understanding is that the grooming behavior reflects the general social function of contact-based chemical communication in cockroach groups. Individuals in close contact exchange cuticular hydrocarbons, and this exchange may help regulate immune activation or stress responses in the recipient. Whether this constitutes care in any morally significant sense is a question biology cannot answer. What it can measure is the outcome. Cockroaches that received increased grooming during illness showed higher survival rates in controlled conditions than equally ill individuals that were isolated. The touch of a nestmate translated into survival probability is a finding that sits quietly at the edge of what we expect from an animal we typically discuss in the context of extermination. The burying beetle Necrophorus vespiloides, already encountered in this script for its elaborate cooperative parenting, has been documented in field studies maintaining stable associations between the same male-female pairs across multiple breeding events. In a species where most reproductive partnerships are transient, some pairs repeatedly locate carcasses together, cooperate in burial and brood care, and show preferential association with their previous partner when multiple potential mates are available. Whether this constitutes pair bonding in the sense the term carries when applied to birds or mammals, implying some form of recognition, preference, and sustained mutual investment, is something researchers are careful about claiming outright. The evidence is behavioral, not cognitive. The same two beetles work together again and again and again. What drives the reassociation is unclear. Shared chemical familiarity may reduce the transaction costs of initial cooperation, or the pair may simply keep finding each other at the same carcasses in overlapping home ranges. Scientists still debate the mechanism, and the debate itself is interesting because 40 years ago, no one would have considered the question worth asking about a beetle. The asking of the question is itself a form of progress. We are learning slowly, and with appropriate caution, that the lives of small animals may be less simple than the lives of small animals were once assumed to be. Stage 4 begins here, and the first fact of legacy and time asks something of the listener, not action, not conclusion, but a particular quality of attention. The Carboniferous period, which stretched from approximately 359 to 299 million years ago, was a world of vast coal-forming forests, oxygen levels significantly higher than today, and insects of extraordinary size made possible by that oxygen richness. Cockroach-like insects, not precisely modern cockroaches, but their near ancestors so similar in body plan as to be immediately recognizable, were among the most abundant insects in those forests. They scavenged the forest floor of a world without flowering plants, without grasses, without birds, without any mammal. The trees were giant ferns and club mosses. The only vertebrates on land were early amphibians and the first reptiles. And moving through the leaf litter, beneath those ancient trees, flat-bodied and quick, were animals whose descendants now live in your city. The dinosaurs would not appear for another 100 million years. The first flower would not open for another 150 million. The cockroach was already there, already perfected, already doing exactly what it does now, processing the dead, recycling nutrients, persisting. When we say a lineage is ancient, we usually mean thousands of years. The cockroach asks us to mean something else entirely. The oldest confirmed beetle fossils date to approximately 270 to 300 million years ago, from Permian deposits in what is now Russia and Germany. These early beetles already possessed the defining feature of the order, hardened elytra covering membranous hindwings, meaning the fundamental beetle body plan was established before the age of dinosaurs began. What is remarkable about the beetle fossil record is not simply its age, but its stability. Beetles found in 100 million year old Cretaceous amber are in many cases assignable to modern families with only minor anatomical differences. A beetle preserved in amber alongside a dinosaur-era flower looks to a non-specialist essentially like a beetle you might find under a log this afternoon. The body plan elytra chewing mouthparts, 6 legs antennae has been retained across more than 200 million years of continuous diversification. What changed was not the architecture but the tenancy. The same fundamental design was repurposed again and again for new ecological roles as environments shifted, continents moved, and ecosystems collapsed and rebuilt. The beetle form is a platform, not a blueprint for a single solution. It is, in the language of engineering, a modular system, the same chassis fitted with different tools deployed across every habitat the planet has produced in the last 250 million years. 252 million years ago, the Permian mass extinction eliminated approximately 96% of all marine species and 70% of terrestrial vertebrate species in what remains the most severe biological collapse in Earth's history. Its cause, or more accurately its causes, involved massive volcanic activity in what is now Siberia, catastrophic greenhouse warming, ocean acidification, and the collapse of atmospheric oxygen in the deep sea. The cockroach lineages of the Permian were not spared. Many of the cockroach-like forms that had dominated Carboniferous forests disappeared, entirely replaced in the Triassic period that followed by the more derived lineages that gave rise to modern cockroaches. Early beetle diversity also contracted severely. What emerged from the extinction was not a continuation of what came before, but a reshuffled deck surviving lineages, radiating into ecological roles vacated by the dead, diversifying rapidly in the ecological space that opened when most of their competitors were gone. The great beetle diversification that eventually produced 400,000 species began in significant part in the aftermath of catastrophe. This pattern, mass extinction followed by explosive diversification in survivors, has repeated across the history of life, and the cockroach and beetle lineages have lived through it more than once. They did not survive the Permian because they were lucky. They survived because the qualities that make them seem indestructible to us were already ancient and proven when the world ended the first time. The dung beetle, Scarabaeus, Satyrus, and its relatives were among the most sacred animals in ancient Egyptian religion, a status that persisted for more than 3,000 years across one of the most sophisticated civilizations the ancient world produced. The Egyptians observed the dung beetle rolling its ball across the sand and saw in that image a correspondence with the sun moving across the sky, both spherical, both traveling in a purposeful arc, both disappearing below the horizon, returning. The scarab became Khepri, the god of the rising sun and of transformation, the force that pushed the sun through the underworld each night and returned it to the living each morning. Scarab amulets were placed over the hearts of the dead in the mummification process, inscribed with spells from the Book of the Dead intended to prevent the deceased's own heart from testifying against them in the judgment of Osiris. The heart scarab was one of the most important funerary objects in Egyptian burial practice, a beetle pressed against the center of a human chest to carry the soul through death and into what came after. The Egyptians who built monuments intended to outlast time itself chose as their symbol of eternity and renewal, not a lion, not an eagle, not any of the large and powerful animals they certainly observed, but a small beetle rolling dung across the desert floor. They understood something about persistence that we are only beginning to recover. The Roman writer Pliny the Elder, in his encyclopedic work Naturalis Historia, completed in 77 CE, described cockroaches, which he called blattae, as creatures that avoided light and infested warm kitchens and grain stores, and recommended several remedies for their removal, including the application of certain plant extracts and the strategic use of cats. This is among the earliest recorded documentation of cockroaches as domestic pests, and it reveals something significant by the 1st century of the Common Era. The cockroach-human cohabitation dynamic was already old enough to have generated not just complaints but a literature of remedies. Pliny's recommended botanical treatments hellebore, certain aromatic herbs foreshadowed 2,000 years of pest control development, in which the fundamental challenge remained unchanged. The cockroach adapts faster than the remedy. Roman grain stores, bathhouses, and bakeries provided the same warm, food-rich, sheltered environments that modern cockroaches occupy in apartment buildings and restaurant kitchens. The ecology of the problem is identical across 2 millennia. What has changed is the scale of the chemistry deployed against it and the growing scientific recognition that the cockroach's resistance to that chemistry is not an accident, but the outcome of one of the most sustained and intensive selection experiments in biological history, conducted by humanity without intending to run it. Throughout medieval Europe, ground beetles and their relatives were incorporated into folk medical preparations with a regularity that modern pharmacology has, in some cases, partially vindicated. Preparations made from crushed blister beetles, family Meloidae, were applied to skin conditions used as vesicants to draw out infections, and administered internally in carefully controlled doses for conditions ranging from urinary complaints to fever. The active compound responsible for these effects is cantharidin, a defensive chemical produced by blister beetles that causes severe tissue damage and blistering on contact with skin or mucous membranes. In large doses, cantharidin is acutely toxic. In precisely calibrated small doses, its tissue-irritating properties were used with mixed success in legitimate medical practice well into the 19th century. Cantharidin remains a subject of active pharmaceutical research. Its ability to inhibit certain protein phosphatases has made it a candidate compound in cancer research, where its capacity to trigger programmed cell death in tumor cells has been investigated in laboratory settings. The medieval practitioner who ground up a blister beetle and applied it to a growth was working without any understanding of the mechanism. The mechanism was there regardless. Historians of medicine sometimes observe that folk pharmacology, accumulated over centuries of empirical observation, contains a higher proportion of genuinely active compounds than its reputation for superstition might suggest. The beetles knew. The people who used them did not know why they knew. The distinction mattered less than the outcome. Domestic beetles family. Domestidae, the hide beetles and carpet beetles, feed on dried organic material, leather, hair, feathers, dried meat, and the desiccated flesh of preserved carcasses. In modern forensic science and natural history museum preparation, dermestid colonies are maintained specifically for the purpose of cleaning skeletal remains. A carcass placed in a dermestid colony is stripped to clean bone with a thoroughness and precision that no mechanical or chemical method fully matches. In ancient Egypt, the relationship between domestic beetles and human remains was less intentional and more complicated. Archaeological analysis of mummies has found dermestid damage on improperly sealed remains, indicating beetle access during or after the mummification process. Some Egyptologists have proposed that dermestid activity on certain mummies may have been allowed or even facilitated during intermediate preparation stages, though this remains debated. What is certain is that the Egyptians were acutely aware of insect activity on human remains. Their elaborate preservation protocols, including natron desiccation, resin application, and tight wrapping, represent a direct technological response to the problem of biological decomposition, beetles included. The mummification process can be read in part as a multi-thousand-year engineering project aimed at creating conditions hostile to the organism's beetles, among them, that would otherwise return the dead to the earth. The beetles, patient as they are, sometimes won anyway. During the Apollo program, cockroaches were discovered in the processing facilities used to prepare spacecraft for launch, specifically in the food storage and equipment areas of Kennedy Space Center. Their presence prompted serious concern among NASA engineers, not because of disgust, but because of a genuine technical risk. Cockroach fecal matter and shed exoskeletons can contaminate sensitive electronic equipment, and the possibility of a cockroach shorting a circuit in a spacecraft bound for the moon was not a scenario anyone wished to test empirically. Intensive pest control measures were implemented throughout the Apollo processing facilities. Whether any cockroaches actually made it into space aboard Apollo missions is not confirmed in the public record, though the question has been raised by historians of the space program. What is confirmed is that cockroach biology was studied by NASA researchers during this period for a different reason: their radiation resistance. Cockroaches can withstand radiation doses approximately 15 times higher than the lethal dose for humans, a consequence of their slow cell division rate during molt, which reduces the window during which radiation can disrupt DNA replication. This radiation resistance, studied in the context of space biology, contributed to a broader understanding of how organisms survive high-energy particle exposure research that has since informed work on radiation therapy, long-duration spaceflight, and the biology of extremophiles. The cockroach did not choose to contribute to space medicine. It simply proved difficult to kill under conditions that kill most other things. Charles Darwin's passion for beetle collecting, documented extensively in his autobiography and correspondence, began during his undergraduate years at Cambridge, where he pursued beetles with an enthusiasm that his academic mentors occasionally found excessive. In one famous passage, Darwin described holding a rare beetle in each hand, and upon spotting a third he did not want to lose, placing one of the first two in his mouth to free a hand, whereupon the beetle released a defensive secretion that caused Darwin to spit it out and lose all three. The incident is recounted as comedy, but the obsession it illustrates was serious and sustained. Darwin collected hundreds of beetle species, contributed specimens to the leading beetle taxonomists of his day, and developed through that process an intimate familiarity with the variation within and between beetle species. The small, consistent differences between closely related forms that occupy slightly different habitats, feed on slightly different resources, or live on different islands. Historians of science have argued with considerable supporting evidence that this early and exhaustive exposure to beetle diversity was formative for the development of natural selection theory. The pattern of variation Darwin later described in finches and tortoises on the Galapagos was a pattern he had already seen in meticulous detail in beetles across England. He knew what adaptive variation looked like before he knew what it meant. The beetles taught him to see. The Galapagos taught him to understand what he had been seeing. Franz Kafka's 1915 novella Die Verwandlung, known in English as The Metamorphosis, opens with its protagonist, Gregor Samsa, waking to find himself transformed into a large insect, described in the original German as an Ungeheuers, Ungeziefer, a monstrous vermin. Kafka never specifies the species, and he reportedly objected to a proposed illustrated cover depicting the transformed Gregor, explicitly insisting that the insect should not be shown directly. The ambiguity was deliberate. Entomologists and literary scholars have nonetheless speculated at length about which insect Kafka had in mind, with the most commonly proposed candidates being a large cockroach or a dung beetle, both creatures associated in the cultural imagination of early 20th century Prague with domestic pollution and social disgrace. The case for the cockroach rests on the description of Gregor's flat body, his many legs, and his ability to traverse walls and ceilings, capabilities of cockroaches but not beetles. The case for the beetle rests on certain details of the shell-like back, Gregor describes. Scientists still debate the question without resolution, which is fitting. Kafka's creature was never meant to be identified. It was meant to be recognized by anyone who has ever felt themselves to be, in the eyes of the people they love, something incomprehensible and unsettling and difficult to look at directly. The insect was a mirror. The species was never the point. In traditional Chinese medicine, the dried and processed American cockroach, marketed under the name Periplaneta americana extract, or commercially as Kangfuxin liquid, has been used for centuries to treat wounds, burns, and gastric ulcers. What was once categorized as folk remedy without biochemical basis has attracted serious pharmaceutical attention in recent decades. Clinical trials conducted in Chinese hospitals have found that cockroach extract preparations accelerate wound healing, reduce inflammation, and promote tissue regeneration at rates statistically superior to placebo controls. The active compounds responsible are still being characterized, but researchers have identified growth factors, antimicrobial peptides, and polysaccharides within cockroach tissue extracts that individually show measurable biological activity in cell culture and animal models. A compound derived from cockroach extract received approval from China's National Medical Products Administration for use in treating traumatic wounds and surgical incisions. Western pharmaceutical researchers have approached these findings with the caution appropriate to a complex biological extract, isolating and testing individual compounds rather than the mixture. But the underlying direction of inquiry is no longer considered fringe. The cockroach that Pliny the Elder was trying to remove from Roman kitchens 2,000 years ago is now, in carefully processed form, being applied to human wounds in hospitals. The distance between pest and medicine, it turns out, was always a matter of preparation. Across sub-Saharan Africa, where cattle herding has been practiced for more than 8,000 years, dung beetles have performed a function so practically essential that their cultural significance developed independently of and long before the Egyptian scarab tradition. A single cow produces roughly 10 to 15 kilograms of dung per day. Without rapid burial and processing, cattle dung accumulates on pasture land, smothers grass, harbors parasites, and reduces the carrying capacity of grazing land with measurable speed. Dung beetles bury that material within hours of deposition, aerating the soil, distributing nutrients through the soil column, and destroying the surface habitat of parasitic flies and worms that complete their life cycles in exposed dung. Studies conducted in sub-Saharan African pastoral systems have calculated that dung beetle activity saves cattle herding economies tens of millions of dollars annually in reduced parasite burden alone, without accounting for soil, fertility improvements. In communities where cattle represent both livelihood and cultural wealth, the beetles that protect that wealth acquired, over centuries of observation, a significance that did not require mythology to sustain. They were simply seen to matter. The practical and the sacred are in the history of human attention to animals rarely as separate as the categories suggest. We tend to make sacred the things we cannot afford to lose. The diabolical ironclad beetle Phloiodes diabolicus lives under the bark of oak trees in the western United States and is preyed upon by birds and lizards that attempt to crush it. Its defense against crushing is purely structural, an interlocking system of jigsaw puzzle-shaped joints connecting its elytra to its thorax and abdomen that distributes compressive force across the entire body surface, rather than concentrating it at any single point. The beetle can withstand compressive forces equivalent to 3,9,000 times its own body weight, without fracturing a performance that places it among the most crush-resistant organisms ever measured relative to body size. Engineers at the University of California, Irvine, published a detailed structural analysis of these joints in 2020 and used the geometry to design interlocking fasteners for joining dissimilar materials, specifically the carbon fiber composite panels used in aircraft fuselages, which had previously required metal bolts that concentrated stress and created failure points. The beetle-inspired joint-distributed load the way the beetle's body does. not by being stronger at the connection point, but by spreading the force so widely that no single point bears enough to fail. The paper describing this work appeared in the journal Nature and was followed by engineering patent filings. The beetle had been solving this problem under oak bark for millions of years. It took a scanning electron microscope and a mechanical engineering team to understand why the solution worked. At the end of the Pleistocene epoch, approximately 10 to 12,000 years ago, the large mammal fauna of North and South America, Australia, and parts of Eurasia collapsed. Mammoths, mastodons, giant ground sloths, glyptodonts, and dozens of other megafauna species disappearing within a geologically first, brief window. The causes of this collapse, human hunting pressure, climate change, or some combination, remain debated among paleontologists. What is less debated is the ecological cascade that followed. Dung beetle communities across these continents had co-evolved with megafauna, specifically with the enormous quantities of dung those large animals produced. The dung of a mammoth or a giant ground sloth represented a resource of a scale and composition that no surviving animal could replicate. When the megafauna disappeared, the dung beetle species, specialized for large mammal dung, disappeared with them. A secondary extinction wave documented in the fossil record of beetle remains preserved in ancient lake sediments and peat bows. Modern dung beetle communities in the Americas and Australia show measurable gaps in species diversity that correspond precisely to the body size range of the extinct megafauna. The grassland ecosystems of these continents are still 10,000 years later running on a dung beetle community that is missing its largest and most ecologically significant members. The loss echoes forward through time in the soil chemistry of every prairie and savanna where the great animals once moved. Among the most scientifically significant cockroach fossils are those preserved in amber fossilized tree resin from deposits in Myanmar, France, and the Baltic region, some dating back more than 100 million years. Within these amber deposits, researchers have found cockroaches preserved not individually but in clusters, multiple individuals of the same species, trapped together in a single resin flow, in postures and spatial arrangements consistent with aggregation behavior rather than accidental co-occurrence. These clustered fossils represent some of the oldest direct physical evidence of social aggregation in any insect lineage, predating the evolution of the social Hymenoptera. The ants and bees and wasps, whose complex sociality has attracted far more scientific attention. The amber captures a moment, a group of cockroaches resting together in a tree, hollow or bark crevice, overwhelmed by resin and preserved in the posture of their ordinary lives. They were not doing anything remarkable when they died. They were simply together as cockroaches have always been drawn by the same chemical signals that draw them together today in the same configuration that the same chemistry produces now. 100 million years of aggregation frozen in resin, waiting in a museum drawer to be examined under a microscope by researchers who are only beginning to ask the questions that the fossil can answer. Flowering plants' angiosperms first appeared in the fossil record approximately 140 million years ago during the early Cretaceous period. Bees, their most celebrated modern pollinators, did not appear until roughly 100 million years ago, leaving a gap of approximately 40 to 50 million years during which flowering plants required pollination but bees did not yet to exist to provide it. The pollinators that filled this role were in significant part beetles. Coleoptera had been visiting flowers, or rather the proto-flowers, of early angiosperms, since before those flowers had fully evolved their modern form. Beetles are messy pollinators by comparison to bees. They consume pollen as food rather than collecting it, deliberately defecate on flowers, and damage floral tissue in ways that bees do not. Early flowering plants evolved in response to beetle visitation, producing robust bowl-shaped flowers with thick, tough petals that could withstand beetle feeding and generating pollen in quantities large enough that some could be consumed without preventing pollination. This syndrome, thick petals, large quantities of pollen, simple bowl shape, often strong fermenting scent, is still called cantharophily, from the Greek for beetle, and it persists in several ancient plant lineages today. The magnolia family, among the oldest flowering plants, is pollinated primarily by beetles. Every magnolia blossom that opens in spring is doing so in the form that beetle feeding pressure shaped over millions of years, before the first bee existed to offer a cleaner alternative. 66 million years ago, an asteroid approximately 10 kilometers in diameter struck the Yucatan Peninsula of what is now Mexico, releasing energy equivalent to billions of nuclear weapons and triggering a global environmental catastrophe. The immediate effects vaporized rock global firestorm. Months of darkness from ejected debris blocking sunlight eliminated approximately 3/4 of all species on Earth, including all non-avian dinosaurs. The cockroach survived. The reasons are multiple and mutually reinforcing. Cockroaches, being small, required far fewer calories to sustain themselves than large animals. Their omnivory meant they could shift dietary sources as normal food chains collapsed. Their tolerance for radiation, documented in modern experiments, may have provided some protection during the intense particle radiation of the immediate post-impact period. Their ability to slow metabolism and reduce water loss allowed them to persist through the extended cold and darkness that followed. Their eggs, sealed inside resistant oothecae, could survive conditions that would kill adults, and their tendency to live in soil under bark and in deep crevices placed them physically in the most protected microhabitats available during a period of global surface devastation. The cockroach did not outlast the dinosaurs through any single extraordinary adaptation. It outlasted them through the accumulated redundancy of a survival system built across 300 million years of prior catastrophes, each one selecting generation by generation for exactly the qualities that the next extinction would require. The specific visceral disgust response that many people experience toward cockroaches is not universal across human cultures, and it is not ancient in the form most Westerners recognize it. Anthropologists studying disgust across cultures have found that the intensity of cockroach aversion varies significantly by region, urban density, and historical exposure. Populations with long traditions of insect consumption show measurably lower disgust responses to cockroaches than populations without such traditions. The intense categorical revulsion common in industrialized Western cultures appears to have intensified during the 19th and early 20th centuries, coinciding with the development of germ theory and the subsequent association of cockroaches with disease transmission. The association is not entirely without basis. Cockroaches can mechanically transport pathogens on their bodies and in their feces, but the degree of danger they represent to healthy people in clean environments is considerably lower than cockroaches. Popular perception suggests what germ theory produced in cultural terms was an emotional intensification of a practical concern. Cockroaches became not merely a nuisance, but a symbol of contamination, poverty, and moral disorder, freighted with meanings that far exceeded their actual biological threat profile. Scientists studying the ecology of disgust note that this emotional amplification, once established, becomes largely self-sustaining, transmitted culturally rather than derived fresh from each individual's experience. We inherit the fear. We rarely examine where it came from, or whether the inheritance is proportionate to what it describes. In the 1960s, Australia faced a mounting ecological crisis. Introduced European cattle were producing dung at a rate that native Australian dung beetles evolved alongside marsupials, whose dung has a different composition, could not process. Pasture land across southeastern Australia was being smothered. Fly populations breeding in undisturbed dung pats reached plague proportions. The economic and ecological damage was measurable and accelerating. The response was one of the more remarkable deliberate ecological interventions of the 20th century, the Australian Dung Beetle Project, which between 1965 and 1985 imported and successfully established more than 40 species of dung beetles from Africa and Europe, specifically selected for compatibility with cattle dung and Australian climate zones. The results were documented and significant. Fly populations dropped, pasture productivity recovered, soil aeration and water infiltration improved measurably in areas with established beetle populations. The project is now studied as a model of biological control, the deliberate use of one organism to manage the consequences of another, and its lessons have been applied to grassland restoration programs in South Africa, Argentina, and the American Southwest. The dung beetle did not ask to be transported across hemispheres and deployed as an ecological repair tool. It simply did in Australia exactly what it had always done in Africa, buried dung, aerated soil, and made the land above it more capable of sustaining life. The intervention was human, the work was the beetles. When ecologists removed beetles from controlled forest microcosms, sealed experimental plots in which all other organisms were maintained, the effects on decomposition rates were rapid and measurable. Dead wood accumulated, leaf litter built up beyond normal seasonal levels, nutrient cycling slowed, fungal communities deprived of the physical disturbance that beetle boring and feeding provides, shifted in composition towards species less effective at breaking down complex lignin compounds. The soil beneath the litter became compacted and less aerated. Within a single growing season, the absence of beetles produced a forest floor that was by multiple chemical and physical measures less capable of supporting the trees above it. Beetles are not merely participants in forest decomposition. They are, in many forest types, the primary physical agents that make decomposition possible. At the rates forests require boring entry points for fungi, fragmenting large woody debris into pieces that bacteria can access, mixing organic to material into mineral soil through their burrowing. Remove them, and the forest does not immediately die. It slows, it accumulates, it becomes, gradually and without drama, less alive than it was, a quieter, less productive version of itself, running on diminished chemistry, processing its own dead more slowly than it generates them. The beetle does not hold the forest together the way a keystone holds an arch. It holds it together the way breathing holds a body, not dramatically, not visibly, but without interruption, and without the possibility of being long absent without consequence. Stage 5 begins here, and with it a different quality of stillness. Cockroaches sleep, not in the way the word implies. When applied to mammals, there is no eyelid closure, no dreaming documented, no slow-wave neural pattern confirmed, but in a form that meets the biological criteria, a daily rest phase characterized by reduced motor activity, lowered responsiveness to stimuli, and a characteristic postural change in which the antennae droop and the body settles lower to the substrate. This rest phase is governed by a circadian clock, an internal biological timer entrained to light and dark cycles, and it is remarkably consistent. The cockroach's active phase begins at dusk. Its rest phase begins in the hours before dawn. The antennae, which during waking hours sweep the air in continuous arcs, slow their movement and eventually fold partially back along the body. The legs, which in motion carry the animal with extraordinary speed, relax their tension and allow the body to lower. The cockroach in rest becomes smaller than it seems when moving more, still more compact, pressed close to whatever warm surface it has chosen for the night's remainder. It does not choose consciously. It finds warmth, finds contact on at least one side, and stops. The doing ceases. What remains is simply the animal breathing slowly through sealed spiracles, waiting, without knowing it is waiting for the darkness to return and the day's work to begin again. Below approximately 7 degrees Celsius, the cockroach enters a state of cold stupor, not true hibernation, but a significant depression of all metabolic and neural activity that leaves the animal motionless and unresponsive to stimuli that would normally trigger immediate escape. This cold torpor is reversible. Warming the animal restores normal function within minutes with no lasting damage, provided the cold was not prolonged enough to cause ice crystal formation in the tissues. The cockroach does not seek cold. It seeks warmth with a persistence that reflects in behavioral terms how thermally dependent its physiology is. In a building, this means wall voids near heating pipes, the motor housings of refrigerators, the warm exhaust spaces behind ovens. In the natural world, it means deep leaf litter, the interior of rotting wood, the chambers of termite mounds, anywhere the earth itself holds enough heat to keep the cockroach's chemistry running through the cool of the night. The search for warmth is not comfort-seeking in any sense that implies preference. It is survival-seeking translated into movement ending only when the body finds what its chemistry requires. And in that stillness pressed against warm wood or warm pipe antennae, slowed body lowered, breathing measured and infrequent. The cockroach rests in the way all living things rest when what they need has been found, and the urgency of finding. It, for a few hours, goes quiet. Many beetle species overwinter in a state called diapause, a hormonally regulated suspension of development and metabolic activity that is physiologically distinct from simple cold torpor. Diapause is not merely the consequence of being cold. It is a programmed state initiated in response to environmental cues, shortening day length, dropping temperature, declining food availability that trigger a hormonal cascade, suppressing development, reducing metabolic rate to a fraction of its active level, and preparing the body for an extended period of stillness. A beetle in diapause may reduce its oxygen consumption by 90%. Its digestive system becomes inactive. Its reproductive development pauses at a precise stage, held in suspension until the hormonal signal to resume arrives with the lengthening days of late winter. The beetle does not experience the passage of the winter months in any way that resembles endurance. It is simply not running. The chemistry that constitutes its life is ticking over at the lowest possible rate, conserving the energy stored in fat bodies through the summer's feeding, waiting for a signal it has been waiting for, in one form or another, for 200 million years. Somewhere beneath the frozen ground inside a seed capsule, under a log's bark, curled in a soil chamber of its own, making the beetle waits not patiently, because patience requires awareness of time, but completely, in the way that only a living thing that has learned to make itself almost nothing can wait. During rest and diapause, the beetle's breathing pattern becomes intermittent to a degree that in a mammal would indicate respiratory failure. The spiracles, those small lateral openings through which gas exchange occurs, remain closed for extended periods, then open briefly in a coordinated flush, then close again. This discontinuous gas exchange cycle, already introduced in the context of cockroaches in Fact 7, reaches its most extreme expression in resting and diapausing beetles, where closed periods can extend for hours. The purpose is water conservation. Every open spiracle is a window through which moisture escapes, and in cold, dry conditions, the metabolic demand for oxygen is low enough that the animal can afford to breathe rarely. The resting beetle holds its breath the way a careful sleeper holds a thought, not rigidly, not consciously, but with a discipline built into the body, over generations of selection for exactly this kind of economy. Between those long closed intervals and the brief opening that follows, something passes through the spiracle that is neither fully held nor fully released, a controlled transition, a measured exchange, the minimum necessary movement between the animal and the air around it. It is in the most literal biological sense the sound of a beetle almost not breathing, and in a forest in winter that almost silence is everywhere, just beneath the frost line waiting for spring. The folding of a cockroach's antennae during rest is not random. High-speed video analysis of resting cockroaches shows that the antennae are drawn back along specific pathways, the same pathway repeatedly across multiple rest cycles, and held in a characteristic lowered position that differs measurably from the alert forward-sweeping posture of the waking animal. The folding involves active muscle control, not passive relaxation. The muscles that position the antennal base continue to fire in a low-level maintenance pattern during rest, holding the antennae in their resting position rather than simply allowing them to fall. This suggests the resting posture is a chosen state actively maintained rather than the mere absence of waking movement. The antennae during active waking hours are the cockroach's primary sensory instruments. They sample air chemistry, detect air movement, assess surface texture, measure humidity, and transmit tactile information about the immediate environment in a continuous stream that demands significant neural processing. Drawing them back into the resting position represents, in functional terms, a partial disconnection from the sensory world, a narrowing of the information stream to something the resting animal can process at reduced speed. The cockroach, in folding its antennae, is doing something that has no precise human equivalent, but rhymes distantly with the act of closing one's eyes, pulling the world a little further away, letting the night continue without it for a while. A dung beetle that has finished rolling its ball for the day does not simply stop wherever it is and rest at random. Observations of nocturnal dung beetle behavior in open grassland environments have documented that resting individuals orient their bodies in the direction of anticipated dawn, aligned within a few degrees toward the eastern horizon, in a posture that positions them to resume their sun compass navigation the moment light becomes sufficient to use it. This orientation during rest has been confirmed across multiple species and multiple geographic locations. Whether it reflects active orientation behavior at the moment of settling or a passive consequence of their final rolling direction is not entirely resolved, but the consistency of the alignment across individuals and sites argues for something more than coincidence. The beetle rests still and dark in a field that is also still and dark, pointed toward a sun that will not rise for hours. It does not know it is waiting. It does not experience the hours between dusk and dawn as an interval. It simply is oriented, positioned, ready in a way that requires no readiness, because the readiness is structural, built into the angle of the body against the ground, written in the direction of a tiny sleeping animal pressed close to the earth. It has spent its day serving up beneath a sky full of the stars it uses to find its way, pointed toward the light it will follow when the light returns. The larva of the stag beetle spends between 3 and 7 years inside a decaying hardwood log, feeding slowly through the seasons, growing through a series of molts, carrying within its body the dormant imaginal discs that will eventually become the adult. During winter months, the larva enters a state of reduced activity, not diapause in the strict hormonal sense, but a cold-driven metabolic slowing that suspends feeding and growth for months at a time. The larva does not experience those months. It does not wait through them. It simply exists at a lower temperature, processing less, becoming less until warmth returns and the chemistry accelerates again, and feeding resumes, and another fraction of a year is added to the slow, patient accumulation of mass and potential that will eventually produce an adult. Somewhere in an old oak log in a European forest right now, there is a stag beetle larva that has been inside that log for 4 years. It has never seen light. It has never moved more than a few centimeters in any direction. It has no awareness of itself, no sense of time passing, no knowledge that it is becoming something, and yet it is slowly in the dark, in the cold, through the patient work of chemistry and the quiet accumulation of years, becoming the animal that will emerge one summer morning, open its wing cases for the first time and take to the air for a life that will last only a few weeks. All of that waiting, all of that becoming, and then briefly flight. In any temperate or tropical forest, in any hour, between midnight and 3 in the morning, the ground beneath the trees is alive in ways that daylight obscures. Cockroaches move along the root surfaces, following chemical trails to food sources established on previous nights. Their cerci reading the substrate for vibration, their antennae reading the air for odor, their compound eyes gathering what little light filters through the canopy. Ground beetles patrol the leaf litter in slow, deliberate sweeps, hunting springtails and mites, and fly larvae with a patient thoroughness that covers every square meter of their territory across the course of a night. Dung beetles navigate by starlight and Milky Way, rolling their loads across open ground in straight lines that would be impressive in daylight and are in darkness extraordinary. The forest floor at midnight is not resting. It is being maintained, cleaned, aerated, decomposed, redistributed by a community of animals that does its most important work in the hours when almost no one is watching. The trees above depend on this work. The soil chemistry that feeds those trees is produced in significant part by what happens in the dark below them. The forest we see in daylight, tall, green, alive, is in a meaningful sense the product of what cockroaches and beetles do when the light is gone and the forest floor belongs entirely to them. The studies that have attempted to measure what ecosystems lose when cockroaches and beetles are experimentally removed tell a consistent story told in the language of soil chemistry and decomposition rates and nutrient cycling and plant productivity, a language that is precise without being, dramatic, and that documents loss without announcing it. Nitrogen cycling slows, phosphorus availability in the soil declines, Decomposition of complex organic matter accumulates at the surface rather than being incorporated into the mineral soil below. Seedling establishment rates drop in areas where beetle boring and burrowing no longer prepares the substrate. Fly and parasite populations rise in areas where dung is no longer rapidly processed. The changes are not instantaneous. They unfold across seasons and years, quietly and without spectacle, in the chemistry of a system that is still running, but running wrong, like an engine missing components that were so fundamental to its design that the engineers who built it never thought to document what they did, because the components were simply always there, always working, never absent long enough to make their absence felt. The forest does not announce that it is failing. It simply incrementally produces less of everything that living things depend upon. The cockroach and the beetle were not glamorous contributors to that production. They were foundational ones present at the base of the system doing unglamorous work for so long that the system was built around them without anyone fully noticing until the moment in a controlled experiment or a disrupted ecosystem when they were gone. 300 million years is a number that resists being held. It is too large for feeling, too abstract for the body to locate in any sensation it recognizes as real. But perhaps it can be approached this way. Every city that has ever existed, every language that has ever been spoken, every border that has ever been drawn and redrawn and drawn again, all of it fits inside the last 0.1% of the time the cockroach has been alive on this planet. Every human civilization, every war, every monument, every extinction we have caused, and every one we have tried to prevent, all of it, compressed into a sliver at the very edge of a history so long that our presence in it is less than a footnote. The cockroach was in the Carboniferous forest before the forest became the coal that powered the Industrial Revolution that built the cities in which the cockroach now lives beside us. The beetle was pollinating the first flowers before flowers were common, before bees existed to take that role more elegantly, before anyone was watching. They did not need us. They do not need us now. What the evidence suggests carefully, without drama, in the language of soil samples and decomposition studies and population ecology, is that we need them. That the systems we depend upon for breathable air, fertile soil, clean water and functioning forests were built over hundreds of millions of years with cockroaches and beetles as load-bearing members. And that, what we call an infestation, what we spray and trap and poison without a second thought, is in fact the oldest continuous workforce on the planet, doing, in the walls of our buildings and the floors of our forests, and the dark beneath our feet, the quiet and unglamorous and absolutely irreplaceable work of keeping the world alive. They were here before us. With care, with the particular kind of care that comes from understanding rather than sentiment, they might be here long after. The night is theirs. It always has been.